I said microorganisms can be classified based on certain 4 characteristics. What are the 4 characteristics I mentioned? Yes, Rebecca, don't go.
Sir, please, their descriptive nature, how they look like, and then the organization that did the discovery, then the
Okay.
one who did the discovery, and
Okay.
then the geographical area in which the discovery was done. Thank
Okay.
you.
That is very nice. So we said that the description, their nature, their look, their shape can be used to describe them. That's where we have something like cocos. We have something like when we have something like staphylococcus. So the coccus there stand for something. Bacillus, cili means something. So the shape, whether the thing is, the microorganism is spheral in shape, is rod-like in shape, all those things are being used to name the microorganism. That's why we say that they are descriptive. Then, as she said, we also mentioned the scientists that discovered it. So we said a scientist like Escherichia discovered a microorganism. So we call the microorganism Escherichia coli, and so on and so forth. We also looked at the institution that discovered them, and then we mentioned the geographical location at which they are discovered. So those were some of the things we said. So you can see it here. We have the descriptive, for example, Staphylococcus aureus is a grape in a cluster of sphere. So because it looks like a sphere, we call it coccus. The coccus there means that sphere is standing for sphere. The coccus there stands for sphere. Then you can also see something like Bacta. So it is also standing for something. So please, what I want you to do is that look at the various shapes of microorganisms and the names they can be given based on their shapes. Look at them. And then we said the scientist that discovered it is the Escherichia coli, discovered by Theodore Escherick. We have geographical place or location at which they were discovered. And examples are there. So those were some of the things we said. And then we said the order of names. The microorganisms are named based on the following order. This is an order. It has been established. It has been there for years. So we talk about the kingdom. The kingdom they belong to. We go to phylum. We come to class, order, family, genus, species, and then the subspecies. So this is the order in which microorganisms are named. From kingdom, phylum to class, from class to order, from order to family, family to genus, and then species and subspecies. So that is the order. Please make sure you keep it in mind. Is that okay?
Yes, please.
All right. So let's look at the rule of naming or the rule of nomenclature. The rule of nomenclature. It says that use binary names. When you say binary names, what does it mean? Or what does it mean?
Binary means two.
Okay. So if you are saying that you use binary names, what does it mean? Or what do you understand by that? Yeah, Jemima?
Two names.
Okay. So it says that binary names into brackets invented by Linnaeus consist of generic name and then a species epithet. So you can see the example there. Can you see this one? Hello?
Yes, please, we can
Yes,
see.
please, we can see.
Okay, so it says that must be used for all microorganisms. Names of categories at or above the genus level may be used alone, but species and subspecies names may not. In other words, never use a species name alone. So you cannot use, when we say a species, okay, now let me bring the whole thing down. When you are talking about a species, you know, we have a lot of human beings on this earth, right? So the human beings we have on this earth, you realize that though we are all human beings, we vary in character or we vary in certain aspects, right? Please, are you with me?
Yes, sir.
So for instance, when you see an Indian man, you know. When you see a China man, you know. When you see an African man, you know. When you see an American man, you know, or you are not able to tell the difference.
We are able, sir.
Okay.
We are able to tell.
So you see that we can consider the human race as what? A species. Now, under that species, we have subspecies. Okay. Under that species, we have subspecies, which means that the general thing is there, general idea is there. For instance, when we talk about human beings, we are under a kingdom, and then we are under the kingdom called the kingdom what?
Kingdom Animalia.
Kingdom Animalia, which means that we are part of the animal kingdom. But do we call ourselves animals?
No, sir.
No, please.
Yes, because human beings are being classified as what? First class animals. Whether you are first class or you are third class, you are still an animal, right? So, so if you are looking at the kingdom, you realize that even under the kingdom, we have a lot of entities that come under that kingdom. So if you are talking about the kingdom Animalia, which talks about animals in general, human beings are seen as animal. Goats are seen as animal. Cow are seen as animal. In fact, we have a lot of animals you can mention, but we have categories for them. Do you get it?
Yes, please.
We have categories for these animals. In the same way, if we come within the human being as a group, we still have differences. We still have differences. Is that making sense?
Yes, please.
It's like we are making an argument, so we want to make an academic argument. So I want somebody to try to describe or say something small about what I am trying to say when we are talking about having a species and then having a subspace under it. I want somebody to also try to add something small to what I have said so far to see if you people are getting what impression or what I'm trying to send across.
Hello, sir.
Yes.
Hello. Hello.
Yes, I'm with you. Can you hear me?
Yes, sir. Sir,
Yes,
I want
Mr.
to
Emmanuel.
use the.
Okay, Mr. Emmanuel, please let the lady finish, then you come in, please.
I want to use the ape as an example. We all know
Okay.
they are a kingdom, but when you see them, they also have different varieties, and that is where the other species come in. In general, they are all eight, but they have species. They have different categories or types within them.
Okay.
I don't know what. Thank you.
Yeah, that is nice. And Mr. Emmanuel, let's take yours as well.
Okay. What I
From
was
there,
saying
we come
too
to
is
Mohammed.
what I also say is in the Animalia kingdom, we have the animals itself there, and then the animals, we have the cat animals or the cat kingdom. And these cats, when you take that kingdom, you have the sub descriptive or description also there. You can have the normal cats that we have as pets at home. You can have the cheetah. You can have the lion itself. You can have the tiger. They all falls under the kingdom of the cat species. So it's also an example.
Okay. Ramat.
Thank you.
No, Rabia too.
Okay, sir. I'm going to use the toad as an example.
Okay.
We have toad and we have frog. When you see them, they look the same, but they are color.
Okay.
We have some that are white, but within their neck is yellowish. Others too are totally green, like the military uniform. Some too are black.
That is nice. Who else?
Thank you, sir.
You're welcome. There's the last person.
Yes, sir. Like dog. If we take dogs for instance, they are animals, but they are different species of dog. We have the red, we have the local type. There are a lot of species of dog.
Okay. So you see, as we're all saying, we're all arguing based on one thing, right?
Yeah.
Our argument is like we have the general group, and under that general group, we have subgroups. Is that not it?
Please, the one with the microphone on, kindly mute yourself.
Thank you very much. So you realize that the kingdom is a broad and most general category. So for instance, the kingdom we've mentioned animals. So all the examples you were giving, talking about the cat kingdom, talking about the dogs, talking about all those things, you realize that they all come together under one umbrella, which is known as what? The kingdom Animalia. But now, when you go down to the phylum, you realize that this group of organisms, they are within the same kingdom. They share similar basic characteristics, and they have certain things in common. They have certain things in common, but then they have things in common. So you realize that under the kingdom, you have certain group of individuals or entities within that kingdom which have their own characteristics. Another group also have their own characteristics, so they share similar basic characteristics. So there we'll say that we have what you call the phylum. So for instance, when you talk about the phylum, we take kingdom Animalia for example. You can mention that fish is a phylum, birds, they are a group of phylum, and then you talk about mammal, they're also a group of phylum. You get it? So after that, then you come to class. So when you come to class, what are that? Now, if you take the same group, for instance, let's take a group like fish. Okay, we have different categories of fishes. We have what we call the oily fish. We have what we call the shellfish. We have the shark, the what do you call it? The dolphin, and all those things. They are examples of fishes, right? But they are examples of fishes, but they belong to a specific what class? Please, is that okay?
Yes, sir.
So we have the kingdom to the animal. Under the kingdom, we have the
Yes, sir,
phylum.
please.
We have different group of
Yeah.
animals. We come to the class where we have rankings of these animals. Then we go down, then we look at the order. Okay, so talking about the order talks about dividing the class into smaller groups of organisms. You see, that share, just say everything in common. They have similar character. They have everything in common. So you can talk about an example like what Brother Ima was using. Cat kingdom. So under the cat kingdom, you can talk about the lion, where you have, even the lions, you may have different breeds of lions, different breeds. So they are all lions, but then they may have different breeds. Just like we have these monkeys, we have different breeds of monkeys. So you see, they all belong to the monkey group, but they have different what breeds. Is that making sense? Hello?
Yes. Lydia wants to ask a question.
Okay, Lydia.
Yeah. Yes, sir, please. I'm lost. It's like I don't really understand.
Oh. Please, anyone to help us out? Hello? Hello, class.
Hello, sir.
Hello, sir.
Yes. Anyone to help us out, please? Yes, Eunice.
Okay, so for my understanding, when we talk about the kingdom, like it is very broad, including like is very broad, including all. Let's take the animal kingdom for example. It's broad, including different kinds of animals. So you have human being as an animal, you get the goats, the sheep, all included as animal. But when you come to the phylum, they are smaller division of the kingdom. And normally the organisms that are grouped there, they are sometimes grouped according to their body plan or stature, or even how at times they look.
Okay.
Or even some characteristics in them. Some of their bone structures are even used in classifying them into the phylums. And at times you come to the class, for example. So even the kingdom, you can even take the kingdom Animalia. And when you come to the phylum, you have animals like having the same backbone grouped into one phylum. And maybe animals without bones grouped into another phylum. Then when you come to the class, for the class they are also smaller division of the phylum. And that place to the animals there have common characteristics more than the phylum. So they look alike more. For example, we will have the lizard and the fish all having backbone because they belong to the same phylum. But when you come to the class, they will not belong to the same class because their characteristics are not that common. Because the fish will now be in water, while the lizard will be on the earth. So, and the other two, okay, that is it for saying.
Lady, are you okay?
Yes, sir.
Are you
Thank
sure you are
you.
okay? Okay. So,
Yes, sir, I
okay,
am.
that is fine. And then we move on to look at the family. Okay. So, family is also a group of organisms within an order. So, the order, within the order, we also have group of organisms that goes into a specific family. So, a family groups organisms within an order that shares very close structural and genetic characteristics. The word there is what? Very close structural and genetic characteristics. So you realize that it is just a breakdown. Let me use this for example. So human beings, we are all human beings, right? But as I said, we have people we call, these are Americans, these are Africans, these are Europeans, these are Asian, and all those things. But even if you come to the African group, within the African group, we have the East Africans, we have the West Africans, we have the South Africans, and all that, though we are within the same African group. Then you come down again within West Africa. Within West Africa, we have different zones again that we can use to categorize. You come down to even come to the nation. And within the nation, we have different ethnic groups that come together to form the nation. Within that same, how do you call it, ethnic groups, you realize that there are different families that came together to form the ethnic group. Please, is that making sense?
Yes, sir.
Yes, sir.
So it is just a further breakdown of the large group that we are talking about to smaller unit or a smaller component. So from there, you come down to the genus, or what you call the general in plural. It's either you say genus or general. And this is a group of closely related species. It is a group of what? Closely related species. So the species that are very much related, they are close. They are very close. So we call them genus, or we say genus. Now, all these things that I'm saying, I'm just trying to let you understand this 8 points, or is it 9? 1, 2, 3, 4, 5, 6, 7, 8. All these 8 points, that all that we are saying over there is just something broad that we are trying to narrow it down to the smallest component or the smallest unit. So a broad concept that we are trying to narrow it down to a smaller component. Is that okay?
Yes, sir.
Is anyone still confused? So let's move on now. So we said that you should use binary names, and then somebody said that we should use 2 names. So you are going to use the genus name, and then you also use what, the species name. If you have a species and then a subspecies, then you use the species name and then a subspecies. It says that when to capitalize, when are you supposed to capitalize? That is what it means. If you are writing these names of microorganisms, when are you supposed to capitalize? The genus name and above, if you are writing the genus name and above, it's always supposed to be capitalized. You are supposed to capitalize it. You get it? If you are writing the genus name and above, you are supposed to capitalize genus name, and so from genus, you go to genus, family, order, class, phylum, and kingdom. So from genus to kingdom, you are supposed to what? Capitalize. Is that okay? Then it says that the species name is never capitalized. For example, you have the Bacillus anthracis. Bacillus and Tracis. You realize that the Bacillus is what? Capitalized, right?
Yes, sir.
Okay. So when are we supposed to italize? When are we supposed to italize or italicize? Meaning you have to, when are you supposed to write it in italics? So names of all taxa. I think by now you remember what taxa is, right? Do you remember what taxa is?
Yes, sir.
Yes, please.
What is taxa or taxa?
When you classify organisms into hierarchy.
Okay, I want the word hierarchy. That is correct. So names of all taxa, that is kingdom, phylum, classes, orders, families, general, species, and subspecies, are printed in italics. They are printed in italics and should be underlined if handwritten. So if you are handwriting them, you are writing them with your hand, what you do is that you underline it. But if you are typing, then you should leave it in what? Italics. Is that okay? Hello? Hello?
Yes, sir.
Yes. Was somebody asking a question or something? Okay, so let's move on. So you are supposed to italize when you are typing, and you are supposed to underline it when you are handwriting, because you cannot turn the pen to italize on the paper. So you underline it when you are handwriting. And it says that if all the surrounding text are in italics, now listen, if all the surrounding text within which you find that organism is in italics, then the binary name would be what? Non-italic, into bracket, the Roman typeface or underlined. So you make it in the Roman numeral form, or you underline it. So you can see an example here. A common cause of diarrhea is E. coli. Can you see it?
Yes, sir.
So you see
No,
that
sir. Sir, I can't see. Sir, please, I can't see it too.
please, are you seeing my slides, class?
No, sir, please
Sir,
explain
I
it all.
can see. You can zoom it.
Yes, so
You
we,
can
we
zoom it.
can see it. Oh. The slides are there.
In
How
the
do you zoom it?
sir,
1 to 2.
zoom it with your hand.
You can zoom it
Sir,
on your phone or the screen to
sir.
observe it well.
So why is the E. coli? The E is shortened, and what's the full name? Why have they shortened the E?
So as we go on, we'll get to know why some are shortened. But the full name is Escherichia coli. I've been mentioning this microorganism long time. That is it here. Can you see it? The one in red here.
Yes, sir.
So Escherichia coli, and then now it has been what? It has been shortened, or you say that the initial has been indicated. As we go on, you understand why it is like that. Okay.
Okay.
So you realize that all these statements
That's
is
the
in what?
standard name for.
Please, you say?
I say shorten. That is the standard name for referencing, American referencing.
Hey, somebody says that. Say, I think you have not started sharing your screen. Please, kindly go log out the meeting and then log on again. Okay. The person who sent the test, I think Sandra something. Please, I am sharing my screen. So if you are not seeing it, kindly log off and then log in again. You will see it. So you realize that here we have all the whole sentence in what? Italics. So the name of the microorganism has not been italicized, but instead
Your
it
screen
has
is
been
on.
what?
Your screen is on.
My screen is off.
It's not off. Maybe check your
It's not
site.
off.
It's not
Yes,
off.
It's
please.
not off. It's on. It's
It's
perfectly
on.
working.
I
Sir.
was doing it, but now I can't see anything again.
Then it is coming from your side, so rectify it. If not, all of us will be affected.
Yeah.
Some of us we are not seeing anything.
That is what we said. Log out and log in. You'll be able to see. If all of us are not
Yeah,
seeing
it has
it,
come.
we'll all see it, and then
I was
the
doing
lecturer will
the
go
thing,
back.
but it went up. Now it has come back.
Okay, Sandra, that is good. Yes, Vivian, your hand is up. Vivian, your hand is up.
Say,
Yes.
you mentioned some example, but we couldn't. I didn't see where that example is. Please.
Can you see the red ink down here?
Yes.
That was the example I'm mentioning. So I was saying that we are saying that when the whole sentence you are working on is in italics, then you don't have to italize the name of the microorganism, but instead you leave it in block and then underline it. So you see in this case, E. coli has been underlined. Are you okay?
Yes, sir.
All right. Then we said that the person who asked the question, when to use initials. The person who asked the question, are you here?
Yes, sir. Yes, sir.
So it says that a specific epithet must be preceded by a generic name, written out in full for the first time. So if that is the first time you are using the name, so you realize that I have used E. coli here already. It is in full. You get it? So if I am going to be using it again in my writing, the subsequent ones, I can write it in what? The first initial. Just as Mr. Emmanuel was saying, even in referencing, when you are writing a reference, it is advised that the first time you are mentioning that thing, you should mention it in full. You get it? So, for instance, if maybe I am saying that, who asked the question? I want to use the person's name.
Grace Bisa.
So, Grace Bisa 2021 says that microorganisms are very infectious, and then we should be careful the way we play around to make sure we avoid microbial infection. You get it? Here I have mentioned all your name. Now, if I am going to mention the name again, I don't have to go and mention the whole Grace Bisa. I should just say Bisa 2021. Anything I will say now, if I am using your name, I'll just say Bisa 2021. You understand it? So that was what he was saying, for example. But in this case, when you have mentioned the microorganism first and you write it in full, the subsequent mention of such microorganism, you may use what? The initial cap. You have to use the initial for the what? For the genus name. Does that make sense?
Yes, please.
Okay. So as we are saying here, it says that written in first use in the paper. Thereafter, the generic name should be what? Abbreviated to the initial capital letter, provided there can be no confusion with other general used in the paper, provided there will be no confusion. So look at the confusion we are going to talk about here. If you are using Salmonella, you are using Shigella, you are using Serratia, you are using Staphylococcus, you are using Streptococcus, they are all starting with S. You get it? So if all these things are starting with SSS, and you know that it is going to be of a problem for you, then you have to be careful when you use it. That notwithstanding, there is something that can help you deal with that confusion. Is that okay?
Yes, sir.
Yes, sir.
So there you move on to what? The common names. The common names. So we said vernacular or common. The common names should be in lowercase, Roman type, non-italicized. So look at this. You see Streptococcus here, which is a general name or a genus name. Then after that, you realize that we have this one also here, which is also a generic name. So, however, when referring to the actual genus name or above, always capitalize and italize. So you keep it in capital and what? You italize it. When you are writing the generic name and above, you keep it in capital and you italize it. Then we move on to look at the species and then the sub. So we are going to look at the subspecies now. Whose microphone is that?
That is patience. Patience.
Yes, Rebecca, your hand is up.
Sir, please, with the common name, does it mean it is the species name that
Patience, please.
has
Put off your mic.
lowercase?
So you see, please, I'm not getting the question. Hello.
Hello, sir.
Yes, I didn't get a question.
Please, I'm asking for the common name if it is the species name that is referring to the common name, since it is asking us to use the lowercase for it.
So this one, you are asking if the species name is what we are calling the common name.
Yes, please.
That is yes. That is yes. So this is the species name. You get it? Hello?
Yes, please. Yes, please.
So now when you go under the species name, you have what you call the subspecies or what? Seroverse. Now when you go under the subspecies, so you realize that we have a microorganism which have other species under it. So for example, you have something like Salmonella. And then under the Salmonella, you have Salmonella typhi, you have Salmonella paratyphi, you have Salmonella enterica. All these are examples of what? Salmonella, right? Please, are you following?
Yes, please.
So when you come here, you realize that we have Salmonella, which is Salmonella enterica. Have you seen it?
Yes.
Then after that, when we are going to mention it again, you realize that the name has been shortened because it has been mentioned already once here. Can you see it? So Salmonella enterica and then S. enterica. I hope this is okay. Hello?
Hello, sir.
Or you are not seeing anything.
We have seen it, sir. Please.
Okay. All right. Then let's move on. So abbreviations for species and then what subspecies. So if you want to write a species, you want to say species, and you don't want to spell everything, you can just write SP. SP. Is that okay?
Yes, sir.
Is it SCP or SP?
It's SP.
Okay.
That is if you are mentioning a specific species. But if you are mentioning several species, then you make it SPP.
Okay.
So look at this. You have Clostridium species. That is one. So if I see this SP, it means that you are referring to a particular Clostridium, right? And then you have Clostridium species. So if I see this, I should know that you are referring to a group of Clostridium. I hope this also makes sense.
Yes, sir.
Then we move on to look at other abbreviations. So the first one we can talk about is EG. This one I have been using it always, which means what?
Examples.
For example, and we say it comes from the Latin word exempli gratia. Can you see it?
Yes, please.
Then we have IE, which means what? That is or does. IE, that is or does. And it says that it's from the Latin word what? It is. Then we move on to look at which one again. There's one ETC, which talks about etc. etc.
Yes.
Then you can also talk about etal, which means more or and others. So maybe I want to use some people in this class to say that I want to reference some people in this class. So I can say that Bisa Jean, that is Bisa Grace, Akosia Sewa or Safoa Akosia. Then I move on to let me look at your names so that I will use it. Let me look at the participants. So I have Abdul Rahim. Hey, your name is that it? Is it double Abdul Rahim?
Yeah.
Hey, compound name. Okay, so let's say that I'm going to use example. So first I mentioned Abdul Rahim, Abigail, or what is the name? Let me just go for it. Osei. So when you are referencing, I guess you know how to reference. So Abdul Rahim, the first one is your surname, right? And then the second one is your name.
Yes, please.
Okay, so Abdul Rahim A, comma, Osei A, that is Osei Abigail. So Osei A, comma, then I write about 3 or more of your names. Now, if I want to write it again subsequently, I don't have to go and write all like I wrote it at first. So I can say that Abdul Rahim A etal, which means that Abdul Rahim and others. Do you understand?
Yes, sir.
Oh, somebody said no. Who said no?
Say me.
Vivian. I'm saying
No.
that. Who?
Akosia.
Say. How many children do you have?
One.
Oh, you have only one.
Yes.
But your brothers and sisters also have children, right?
Yes.
Yes.
So let's assume that 4 of them come together to write a paper, right?
Yes.
So give me the names of those children.
Alex.
Alex what? Eh?
Boatin.
Okay, so Boatin, Alex.
Bernardina Henkai.
Bernardine or Bernard?
Bernardine.
Okay, so Ahinkai Bernardine.
Yes. Samuel Brobe.
Brobe Samuel.
Francisca.
Francisca what?
Francisca.
So you see the way you have mentioned their names.
Yes.
In referencing, you don't bring the first name first before the surname. You bring the surname before the first name. And then after writing the surname, which is Boatin, you don't write the one which in the one in addition, the first name, you don't write it in full, you make it initial.
Okay.
So you said NS, right?
Yes.
So Boatin, comma E. Yes. Then I bring dot. You know that if you write initial, you have to bring a full stop, a dot in front.
Yes.
Which should tell us that. So Boatin E, comma Boatin, comma E, dot, comma. Then I bring Ahinkai, comma. Then you said what? Bernardine. So B. So it will be something like this. Let's say Boatin.
Vivian, please mute. You are not speaking, so mute.
Yes, sir.
Then you said that Ahinkai. If I don't spell it well, don't laugh at me. It's not my language.
Yes. I won't laugh.
Is that how you spell it?
No.
Like this.
Vivian, please mute.
And the next name.
I Francisca.
No, you mentioned something before Francisca.
Brobe Samuel.
So Brobe.
Comma.
Samuel.
Sam.
Hey,
So I think you said it.
And so the end, I'll bring this sign. You do know it, they call it the ampersand sign. You see it? And that right?
Yes, F. Dot.
Maybe 2024.
Okay.
And then I'll write what they said. You get it? Maybe the title of the article they wrote in. Now, the next time I am going to do this, or I'm going to reference these people, I don't have to write all this again. So I'll be like, Boatin et al. 2024. Can you see it?
Yes.
So the et al. Here, this et al. Means Boatin and others. So and others. So the others are there, but their names are not going to be mentioned. So et al. Means and others. So I'm trying to say Boatin and others. But because they are plenty, I don't want to write all of them again because I have written it first. So I don't need to write it again.
Yes, sir.
I hope it's well understood.
Yes, sir.
Class, do you have any problem? Any question?
Yes, sir, please, today's lesson, there's not going well with me. Because I don't see anything on my platform. I've been logging out, coming in. I don't see anything, so I don't understand you.
But please, I don't think you should only see my screen before you can follow me, because I've already shared this slide with you since last week.
Because it seems what we are doing, we have to see it and follow you.
Sir, serious, today the network, we are not finding it easy at all.
I just listen, but I don't understand anything.
And this one, you see, it is from no fault of mine. I don't know what I can actually do about this one. But then when you take the slides are with you, right?
So yes, I have even printed them before me, but I can't see anything.
Then it means you're actually not following what you are doing, because we are doing the thing point by point. It's only when we get to a point that I'm supposed to explain, then I will just type under the same slides. Like this one I'm doing right now, it's not there, but I'm doing it under the same slide. So if you were following, we are on rules of nomenclature. And this, we are on the 7th rule. So the 7th one, we are just looking at other abbreviations that are being used. Abbreviations
when
that are
you go to the rules of nomenclature, that's where I had a problem, because I wasn't seeing it from the screen. When you started with the rules, that's where I had problems, because I wasn't able to see from the screen.
Okay, so class, what do we do now?
She should minimize what we are doing and then open to the slides from the integrated page.
She said she has already printed it. So I guess if she was even following from her hard copy.
So she will tell that she should minimize the and go through the whole slides and follow it. That's what I was saying.
Hello, sir.
Yes.
The test itself is not easy to understand. The test itself, that is how it is. It's very difficult test. So I think that is one factor. What we are discussing is not that easy.
Hey, naming microorganisms is not easy for you then.
Thank you. I'm even understanding it by your explanation, little by little. So that's why my sister is complaining. The test itself is not easy. It's not in your face.
Say, the printed one that she has printed and open the rules of nomenclature, point 7, abbreviation notation, so that you can.
If you don't understand point 1, how can you understand point 7? So what she's saying, her network, just like my network, is
I think she should look for a convenient place that she will get a full network, and she should log out and come in again. She will get it. She have the printed one. She can follow us through the printed one if she can't use the slide together with the meeting on her phone or laptop. Or better still, if she has the laptop or the phone, she can minimize the meeting and go back to where the slide is and follow us there, because the fault is not from our side, it is from her side. Maybe her network isn't going through.
Please,
so she can log out and
is that look for somebody's phone and use it to rejoin, if any. Please kindly look for another phone, if any, and then log out. Then you send the link to that phone and then use it to join.
Okay, I hear. Thanks. Thanks everyone.
let's continue.
Hello.
Hey.
Hello.
Say.
I'm still on. I don't know what I am doing now. Any suggestion as to how we can go about it?
Just continue. You writing and foundation.
Sir, please let's continue.
Please let's continue. At times it's like that, but when you finish and we sit down to go over it, you are able to get the whole idea.
Okay. So let's move on. I think personally I'm even talking too much. So I'll be calling names. Observe what is on the screen. Observe this. The rule of nomenclature, where we have the plural forms. Okay.
Hello, everyone. Welcome.
So the plural form, we are saying that when we use plural and when we use singular, or if it is plural, how do we mention it? If it is singular, how do you mention it? So we have something like genus.
Sometimes.
We have genus, and then the genus, that's the singular name. The plural name is general. Is that okay? Then we have species or species. So when it is one, we call it a species or a species. And then when it is more than one, you call them species. S is added to it. And then if you want to abbreviate it, it's SPP. Then medium, the plural is media. Okay. Medium, the plural is media. So you realize that we have a lot of medium we have been communicating on. And together we say we call them what? Social media. So they are mediums that we use for communication. Vivian, your hand is still up. What's the problem? So we have fungus, and then the plural is fungi. We have certain examples here. We have staphylococcus for one. When it is small, we have streptococcus for one. When it is small, we say streptococci. Staphylococcus, if it is small, we have staphylococci. Enterococcus, if it is small, enterococci. Bacillus, one. Bacilli, more. Bacterium, one. Bacterial, more. So we don't say bacterials, no, we say bacterial. Immediately you mention bacterial, we know that it is more than one. We have alga.
Portia, you are
Plural,
disturbing us, please.
So when I plant from us today or tomorrow?
We have protozoa.
Portia.
The plural is protozoa.
Oh, sorry.
So now let's take the organisms each one after the other and look at certain things they have. So we're going to look at bacteria. let me mention names for them to read. Adelaide Ba Sapon. Can you see my screen?
Hello, sir. Hello.
Yes.
Yes, please.
Yes,
I'm
Madam
there.
Adelaide, we are hearing you. You can see the screen, eh?
Hello.
Okay, so read what you can see.
Please, under the bacteria.
Yes, please.
Sir, please, am I supposed to read under the bacteria? So
Yes, please.
Bacteria into bracket singular. Bacterium.
Adelaide, we are sorry, your network is helping.
Microscopic single cord organism.
The network is not fair. We can't hear her.
Adelaide, your network is not good. So, hello, Adelaide. Hello.
Every environment on Earth.
She can't even hear you. That's why she's telling me.
Adelaide, please pause and don't read again. Oh. Yeah, someone.
Hello.
Yes. Adelaide, your line was breaking, so you don't worry. I'll call somebody to bail you out. Eh?
Yes, please. Okay, I hear.
Alice, Phyllis. Alice, Phyllis.
Hello. Sir.
Yes.
I should read down the bacteria.
Yes.
Okay, bacteria, singular, bacterium, is any of a group of microscopic single-celled organisms that live in enormous numbers in almost every environment on Earth, from deep sea vents to deep below Earth's surface, to the digestive tracts of humans. Bacteria lack a membrane, bound nucleus, and other internal structures, and are therefore ranked among the unicellular life forms called prokaryotes, right? They constitute a large domain of prokaryotic microorganisms. Bacteria have several shapes, including spherical, rod, vibro, and spiral.
Okay. Please, how do you understand this? Not you, not the one who read it to. Anyone at all in the class. Guys, how do you understand it? Just raise your hand, I'll call you. Alidu, the other one I cannot mention. I'll bite my tongue. Yes, Alidu. Is it Zuwera to or please? I don't want to bite my tongue. So
okay, sir, I'm here.
Okay. What is the name?
Sir, please. Zuera.
Zuera.
Zuera, yes.
Oh, okay. Next time I'll try to mention it.
Yes. If
Okay.
not that, you pour the signs in your mind in my mind.
Hey.
My mind. Okay, sir. It's trying to say that bacteria are everywhere around us. Bacteria are I don't know how to explain it.
I said we don't say bacteria. No, there's nothing like bacteria. It's
yes,
bacterial.
Bacteria. They are everywhere around humans, and even when digestion is taking place, they can be there. Yes, that's how I understand it too. And they further explain the shape of bacteria.
I want.
Okay.
Yes.
I'm done.
I want somebody to explain the second point. This one. This is what I want.
Membrane. Okay.
Yes, who is explaining it? The men are hiding. Let me call the men. Abdul Rahim.
Yes, call them, say.
Yeah, I'll bring them out. Don't worry. I'll fish them out. If I call your name, don't leave the class. You leave the class, I'll charge you.
Sir.
Abdul Rahim, how do you understand this statement? Bacteria lack a memory-bound nucleus and other internal structure, and are therefore ranked among the unicellular life form called prokaryotes. How do you understand it? Abdul Rahim, you are here. Talk. I'm listening. Hey, he's gone. Okay.
Hey.
Ankama Kwabena, please, are you part of us? Or you are an observer? Ankama. Hey, he's also not minding me. Before I realize he'll run away.
They are hiding, sir.
Hey, Dixon. Mr. Emmanuel. You have to save your people.
Yes, I'm here.
Your other gentlemen are not. They are running away.
Mr. You must save your generation. Save them.
Okay. What I understood of this is they said bacteria lack a membrane. I want to talk about memory. Membrane.
So
yes.
Please, Zema, let's focus on this. Put this together. It's memory-bound nucleus.
Nucleus, yes, memory-bound nucleus. So when we talk about memory-bound nucleus and other internal structures, I want to talk about the unicellular form.
Okay.
So when you talk about the unicellular form, it means that it consists of a single cell.
Sure.
Yes, a single cell. And they are also a type of prokaryote, as we said at the last part of it. And it also means that it lacks nucleus and other memory-bound structures. So I don't know how to put it for a very direct understanding of this. Some of these things, explaining it into details will rather confuse more.
Okay.
So
let me come in. Okay. I actually brought up.
I want to when you talk about single structure, or okay.
Yeah, let me come in. Okay. So you see, when we were doing plant cell, animal cell from basic school to secondary school.
Yes.
We have cell membrane, right?
Yes.
And then in the membrane of the cell, we have a nucleus. Within the nucleus, we have certain components. What are the components of the nucleus? Look for it, and then I'll give you the answer to this.
Those are components for
Is that what? Components of the nucleus.
A cytoplasm part of it.
Cytoplasm. Hey.
Sir, I would say the components of the nucleus. We can talk about the presence of DNA.
The DNA.
Yes, sir.
Hey, you people are mentioning big words, so.
The mitochondria.
Mitochondria. People are mentioning big words, Papa. Okay, let's continue. I'm waiting. I'm listening to you.
So you can
chromosomes.
Chromosomes. Hey. Okay, say more.
Cytoplasm.
Cytoplasm.
The nuclear pulse.
What?
The nuclear pulse.
The nuclear pulse. Mm-hmm. Okay.
Membrane. The nuclear membrane. Membrane.
So
with prokaryotes, they lack cell membrane. They lack nuclear membrane.
Okay. So in the nucleus, okay. In the nucleus, we have a lot of things you can talk about. Like you said, the chromosome, we have the DNA, RNA, and all those things are there. But what we are trying to say is that from this explanation, we are saying that these microorganisms, okay, the bacteria, they are prokaryotics. In other words, these microorganisms have a single cell. They are single-celled. And after they being single-celled, their memory, okay, their nucleus is not bounded. When you say bound, like you have something surrounding it, you get it? Hello? When you say you are bounded, it means something is surrounding you that is preventing you to explore or to go to wherever you may want to go to. It is limiting you to a particular level or a particular area, right?
So please, can I come in?
Yes.
To my understanding, just like a baby is formed in a womb, and the baby is also found in a sac inside the womb, the same way the prokaryotic lack membrane. They don't have membrane, anything that bounds them.
So for instance, you don't have the sac, like you are using the baby, for example. There's nothing like the sac that is bounding the bacteria, right?
Yes, please.
Yes. That is the only. Somebody's microphone is giving feedback. Okay. So you see, in the nucleus, we have genetic materials.
Is it 082? Whose microphone is giving the feedback?
Okay, so please kindly try and then. So in the nucleus, we have a lot of genetic materials that are formed in the nucleus. So we are saying that when you are taking bacteria, for example, some people will say that it doesn't have a true nucleus. True nucleus means that it doesn't have a definite shape, a definite described nucleus. Is that okay? So in their cytoplasm, you realize that the genetic material or the DNA you people are mentioning is not bounded by anything. So it floats, it's free, it moves freely within the what? Within the cytoplasm. So that is what we are talking about. So it says that this bacteria, they lack a memory bound nucleus. So it means they have the nucleus that is not memory bound. It is not bound by any memory. And its internal structure and other internal structures. So it means that other internal structures are also not memory bound. Like they don't have anything that is shielding them or surrounding them. They are just moving freely, free range animals. You get it? So that is what it means. So we are saying that bacteria are prokaryotes. In other words, they have single cell and then they can be found everywhere. and they don't have a definite shape memory bound, or they are not memory bound. This one, take it from here. That point, that small point you are seeing, that small, yes, point you are seeing has 3 different individual points that if we ask you to differentiate bacteria from any other microorganism, you can use. Does that make sense?
Yes, sir.
They are
Yes,
unicellular, which means that they are single-celled. They don't have memory bound. They don't have memory-bound nucleus, and then they also what? They don't have memory-bound nucleus. They are unicellular. For that matter, they are prokaryotes. Then when it comes to the shape, that's where I was telling you that we have the spherical shape, and I say if it is spherical, you call it what?
Coccus. It is called coccus.
It's coccus. Okay. What about if it is rod-like? If it is rod-like in shape?
Descriptive.
No. If it is rod-like in shape, what do you call it? I will search for it after this class.
Heptococcus.
Hey, somebody saying coccus. No. So look for it. Look for this 4. When a microorganism is spherical, how do you call it? When it is rod-like, how do you call it? When it is spiral, how do you call it? When it is, we say it's vibro, what does it mean? So look for them and then tell me. But not now. I'll take it maybe before we start.
Helicobacter.
Helicobacter. Okay. So what I want you to do is that take the listing. How do you call it? These ones, take them individually and then search for what they mean. Okay, before you attach them to any microorganism. For instance, let me do this for you to see. So,
so can I come in?
Okay, come in.
So when it is spherical, just as our auntie says,
coccus.
Coccus. And then the rod shape is bacillus or bacilli.
Bacillus or bacilli. Hello?
Yes, sir.
So
the
we said that
bacillus is singular, and the plural
bacillus
is a single.
Sure, that is
and
true.
The bacilli is the plural.
Okay.
Then when it is spherical, it's called spiral. Yeah, so it's a noun. I'm pronouncing it anyhow.
Yeah,
and
don't
then
worry.
the singular is spiralium.
Please, can you hear it?
No, please.
No, please. You can also search for it. Don't worry here. Please, let's continue.
Then when it is,
I do also spell it.
You can also search for this thing. It's not anything. Search for it, you see it.
Mom. So let's continue.
Yes. It is just good you understand it. So search for it and then understand it for yourself. So it says that when you continue, it says that although most bacteria are harmful, even beneficial. Let me just put it in my own sentence for you to understand. So though we are saying that bacteria is harmful, or some bacteria are harmful, some are actually beneficial to us as individuals. Okay, they are beneficial to us as human beings and then living organisms. So it is not all bacteria that are harmful. Do you understand it? Because some bacteria are used in fermentation of food products that we consume. For instance, the lactobacillus, okay, is used in fermentation of milk in the production of yogurt. And if that bacteria is not there, the fermentation process will not be successful, and so you will not get a good yogurt to take. Then we have other ones that are also used in the fermentation of wine, like grape for wine. And we say that they make the drinks or whatever they are using possess a probiotic function, which is good for your gut health. You get it? So we call this group of microorganisms beneficial microorganisms because, in fact, they serve a benefit to us, those who consume them. Is that okay?
Yes, sir.
Yes, sir.
All right. So I can say that some of these bacteria also cause a lot of sicknesses and infections to the individual. So you can talk about the HPV. What is it?
Human papilloma.
No, HPV is actually a virus, so we shouldn't go there. But we have bacteria that causes a lot of infections. So something like Candida. Please mention some for me. Yeah, I've forgotten them all. Hello.
Sir, please, can H. pylori be an example of it?
That is Helicobacter pylori, which causes
Stomach
ulcer,
ulcer.
right?
Yes.
Yes. Is it an enzyme or bacteria? Yeah, it's a bacteria. Yeah, it's part. It's an example. Give me more. Oh, give me more bacteria that causes infections and diseases.
Diarrhea.
What?
Diarrhea.
Diarrhea. Diarrhea is a sickness. It's not a bacteria.
Yeast infection.
Yeast infection. Okay, yeast infection is there.
Sir, please.
We have yeast and mold as a different group of microorganisms, right? Yeast and mold.
We also have fungi.
The fungi is also different, but I want examples of bacteria that causes diseases or sicknesses.
Okay, so sir, we can talk about
bacteria.
What?
Protozoa, is it a bacteria?
Protozoa.
Yes.
It's
Which
also
causes malaria?
It causes what?
Malaria.
Malaria.
Yes.
Protozoa.
And I'm asking if it's a bacteria.
Sure, it is a bacterium.
Okay.
Please, we can also talk about
what?
So what about Samolina?
Please, let me come in. Please raise your hand and allow the lecturer to call you because we have more than 5-6 people talking at the same time. If you are not being called, don't talk so that it will be well arranged for us to have a successful class. Thank you.
Okay. So somebody was mentioning we have something like Salmonella typhi, which causes typhoid fever, right? Linda.
Yes.
Yes.
What?
Yes, sir.
Yes, Linda, your hand is up. I'm listening. We also have Mabla.
We also have tetanus.
caused by
Untimembo.
Is this
I
Clostridium
mentioned the
type?
salmonella type.
You mentioned the salmonella typhi. Okay, that's good. You said it's caused by Clostridium what?
Tetani.
We have
Yes, please. I
Clostridium
mentioned
perfringens. Please let me mention the names for you to see if that is what you want to mention. We have Clostridium aureus. We have Clostridium perfringens. We have Clostridium botulinum. We also have
PETANI.
PETANI.
Clostridium tetani. I-E.
Oh, okay. So it's Pepani.
Oh, okay.
All right.
And then tuberculosis caused by Mycobacterium. Hey, so let's continue.
Now, once you have something like cholera, cholera is caused by Vibrio cholerae. Very simple. You also have something like how do you call it? So we said typhoid fever, and then we have Salmonella paratyphi, which causes paratyphoid fever. That one is also there. You can also talk about
They are all anti-scientific.
they are all what?
They are all big.
They are all big. So learn to mention them. Why? Let's continue. So we are moving on to look at viruses. Now, when you talk about viruses, I'm not going to make anybody read. I'm going to explain it. When you go, you read for yourself. When you talk about viruses are particles that are made up of genetic materials. Now, when you talk about genetic material, you should know what we are talking about. Yes, Alice.
Oh, sorry, I wanted to ask a question, but then I've gotten an answer already. Thank you.
Share with us. Maybe somebody will also want to ask the same question.
It was on the types of bacteria.
Okay.
I was going to ask of the name of the bacteria that causes tuberculosis.
Oh, okay. But now they have mentioned it to you, right?
I went online and I was looking for it. So
okay, so share with your friends.
They said it's Bacterium tuberculosis.
Bacterium
tuberculosis.
It should have an actual name.
Yeah, it is Mycobacterium tuberculosis.
It is what?
MYCO. Then the bacterium.
Please, I don't know if I am the only one who is not hearing.
Sir, the one that you just mentioned. How can a bit to answer bacterium tuberculosis number?
Yeah.
It's Mycobacterium tuberculosis.
Okay. All right, so let's move on. Please, I hope nobody has a question again, or anybody has a question. Is anyone having a question?
No, sir.
Okay. Then let's move on to look at viruses, okay? Now, when we talk of viruses, we said that they are made up of genetic material which is housed inside a protein shell or a capsid. Now, when we talk about genetic materials, all we are referring to is what? The DNA, the RNA, and all those things. So anytime you hear genetic material, it's talking about the genetic makeup of that person. So the gene that comes together to form that person, the genotype, and then the how do you call it? The gene, the genotype, and then we have the genome. So all those things come together to form what we call the genetic material. And we said that the virus, it has genetic material which is housed inside a protein shell, which means that the genetic materials are inside a protein shell. That is how a virus looks like. Genetic material. Yes, someone is talking. So the genetic material or the genome of a virus may consist of a single-stranded or a double-stranded RNA and then DNA, respectively, and may be linear or circular in form. So when you are talking about the DNA and then the RNA, I don't know how much you understand about the DNA and then the RNA, but then you'll be doing much into the DNA and RNA when you come to the second year. I don't do years. When you are doing microbiology and then biochemistry, that notwithstanding, let me take you small into the DNA and then the RNA. So the DNA is actually formed from what we call nucleotides. The nucleotides are also made up of sugars. So the DNA, we say it is called the deoxyribonucleic acid. Okay, the deoxyribonucleic acid. That is the DNA. It has a sugar which is called the ribose sugar. So, and it also has a base. So these nucleotides come together to form what you call the DNA strands. And then when they come to form the DNA strands, that is being used in a way to produce the RNA. So all these things goes on within the individual or within an organism. Now, for viruses, viruses are very infectious. And one thing about viruses is that they have no life if they are outside a cell or a host. They don't exist, or they have no life. They are there, but they are not living when they are outside a living cell. So for instance, you remember when COVID came, they were like, you should wash your hand, you shouldn't be taking your hands to your nose, you shouldn't be putting your hand in your mouth and all those things. Because the virus can be on your skin, it will not do anything to you because your skin has a lining that is protecting it against the cells. But immediately it enters into a cell, a live cell, this virus will start to what? React, or it will start to what? To work, or it will start to destroy. So it says that virus is an infectious agent of small size and simple composition that can multiply only in living cells of animal or plant or bacterial. They pose unique infective property and thus often cause diseases in the host organism. So when they are outside you, they don't cause any problem. But immediately they get a chance to be inside of you as an organism, they will start to cause problem within your system and it will destroy. So we have a lot of viruses that can affect or cause an infection to an individual. For example, you know we have the hepatitis E virus, right?
Yes, please.
And then we have the hepatitis A virus. We also have the HPV, the human papilloma virus. We have a lot of viruses, but all these viruses, when they are outside the cell, they are outside the living cell, they don't work. They are inactive. Immediately they enter into the cell, they become active, and then they start to cause infections to you, the individual. Now let's look at fungi
It's
or
Catherine. Catherine has a question.
Yes, Catherine.
Say,
Yes.
please, I've learned that bacteria they have good side. Does virus also has a positive side or a benefit to the host as we? That's what I want to find out.
Okay, oh, it's a question to all of us. Viruses, do they have a good side?
I don't think they have.
Yes.
Okay, sir. So, looking at the last sentence, viruses possesses unique, ineffective properties and often cause diseases. I don't think they have any good side or any
Yes, they have
positive
good
effect.
sides. They have good sides.
Yeah, what are some of them?
One of the good sides is we normally use the same virus to produce antivirus or to produce medicines.
Yes.
So even though they are used to, they harm us. Even though they harm us, you can still use the same virus to produce antivirus, which fight against other virus. That means that they are regenerated or they are altered to fight against themselves. That is what I want to say.
Okay. Is that who else has a different view? Who else or anything to add? Yes, who else has a different thing to add?
Hello, sir.
Yes.
Please, I've raised up my hand for some time. I'd wanted to ask a question. I don't know if I can put it over.
Oh yeah, you can bring it, please. No, let's finish answering hers, then we come to yours. Okay.
Okay.
All right. So let me just add something small to what he said. So, you want to? Okay, if you want to say something, just say it, please.
I just want to add another one. It can also be used, a virus like oncolytic virus. It can also be used to treat cancer, even though cancer has its own effects. At times, we use a certain viruses to fight these cancers. That keeps humans for a long period of time before it really brings the person down or the host down. Thank you.
Okay, that is very good. And then so actually, I was saying, everything that has its negative side will also have a positive side, no matter what you talk about. So as Mr. Iman said, some viruses are also used to create antiviruses that is used to fight other viruses. You get it? So in as much as the viruses, we say that they are dangerous to our health, some are also used in a way, even in farming or in plant growth. Some viruses are used to fight against other severe viruses from destroying the plant. So for instance, in tomato farming and then in pawpaw farming, they use certain viruses to fight against other severe virus infections towards the plant. And some are even used to produce antibiotics. Is that okay? Some are used to produce antibiotics, and then it is used to kill harmful bacteria that prevent you from what? Absorbing your antibiotics. Please, are you with me?
Yes, please.
Some are also used to control microbial infection within a population or within a group. So they are beneficial. That's the conclusion. They have their own benefits. So I guess you can move on now, right? No, the second question, please.
Sir, when you were talking about virus, you said that virus moves through a medium, and that
Yes.
if the medium is not there, the virus is considered dead. But I read somewhere that viruses never die, rather they get dormant. So a virus like HIV can be in existence for as long as it has not gotten a medium. But the moment it gets a medium, it revives back. I want to know if this is true, then is it because it is within a protein shell, or what gives it that factor? Thank you.
Well, this is a nice question, but then please, I stand to be corrected. Did I use the word dead?
Yes, you used it that it might be considered as good as dead or something of the sort. Yeah.
Oh,
are you sure?
I heard you saying that it becomes dormant, ineffective. That was the word.
Yeah.
Really?
So
Okay.
I don't know what you heard, but then I don't remember using the word dead. I said that they will be there, but they are not active.
Okay.
Mm-hmm.
Yeah, thank you.
All right. So I hope that is okay. Or there's an explanation. Hello.
Sir, please, if they become dormant, will they be active again? And if they will be active, what will cause them to be active?
So you see, from the question you are asking, when the viruses are there, okay, they are inactive. If they are inactive and then conditions like heat, excess heat is applied to them, it will destroy them. You get it? So, for instance, let's say that somebody used a razor or a blade and the person is having HIV virus, okay? And then the person, after using the blade, left the blade for some time. The virus can be on that blade. But if the blade is sterilized by another person before using, the sterilization process will kill the virus.
Okay.
Yeah, it will kill the virus or destroy the virus. So the virus will not be in existence again. But if nothing like sterilization is done and you use the razor, then the virus which is there can now enter into your cell if only you have a cat. And there it will start to work.
Okay.
Please, I hope it is well understood.
Yes, please, I'm asking that if it's in the system and it becomes a body system.
If it does what?
If it's in the body system and it becomes dormant, inactive, what will cause them to be active in the system again?
So far as it is in the system, it can never be dormant.
Okay.
So we said that when it is outside the cell, it is lifeless. But immediately it enters into a cell, it starts working. And you should know that we have factors for microbial growth, okay? We have factors that are responsible for the growth of microorganisms. Some of the factors you can talk about is temperature. You can talk about moisture. You can talk about pH. You can also talk about water activity. That is the same as we have moisture and we have water activity. Then you can also talk about the nutrients. You can also consider, what do you call it? I've mentioned pH, I've mentioned nutrient, I've mentioned water activity. You You can also mention presence of other microbes or organisms. All these things are factors that can enhance the growth of what? Certain microorganisms. Okay. So the virus, for instance, when it is outside the cell, it doesn't have anything to feed on. It doesn't have anything to work on. So it is lifeless. Immediately it enters into the cell, it is going to be feeding on from your cell. So now it becomes active. And as it increases, what happens is that when it is feeding, it is increasing in numbers. Like the way human beings, you eat and grow in size. Microorganisms do not grow in size, but instead they multiply in their numbers. Please, does that make sense?
Okay. Okay.
Yeah. All right. Zuera, I saw your hand up.
Oh, sir, please, it's a mistake.
Okay. All right, so let's move on to fungi. Let me mention somebody to read. Some people are here, they are not minding us. Bet Asari Boatin. We are on fungi. She's not there. Rita, Pokua.
Hello, sir.
Yes.
Yes. Please, should I read?
Yes, please.
Yes, please. Okay, fungus, that's plural fungi of the kingdom fungi, which includes yeast, rust, mats, mildews, and mushrooms. Historically, fungi were included in the plant kingdom. However, because fungi lack chlorophyll and are distinguished by unique structural and physiological features, that is, components of the cell wall and cell membrane, they have been separated. Fungi are eukaryotic organisms, that is, their cells contain membrane bound organelles and clearly defined nucleus. Animals such as cats and dogs have eukaryotic cells. Plants such as apple trees have eukaryotic cells. Fungi such as mushrooms have eukaryotic cells. While mushrooms and toadstools into bracket poisonous mushrooms, hello,
Yes, I'm with you. I'm with you.
are by no means the most numerous or economically significant fungi. They are the most easily recognized. Fungi, other than mushrooms, are sometimes collectively called molds, although this term is better restricted to fungi of the sort separated by bread mold, represented by bread mold.
I projected something for you to read. You are just reading. The rest, where are you reading from? Your book. Okay, so let's discuss. How do you understand this one too? Let me call some few people. How do you understand this one? This one is very simple, so you should be able to Tell me. Rahina too, Idrisu. How do you understand this? Those that if I say I'm coming to call for discussion, but then you are going off. I'm seeing all of you. Rahina is not talking. Patience Okansi.
Ene.
Talk. This one is just discussion. Say what you understand by what you are saying. Yes. Oh, you are not talking. Please, who want to talk? Let's just share ideas, okay?
Something small. Fungi, I understand to be a vast and diverse kingdom of organisms, separate from plants, animals, and bacteria. They play a crucial role in the ecosystem as decomposers. Some can cause diseases in humans and animals and plants. So is it okay?
Yeah, it's okay. So somebody should also add up. All the points are there. You just say how you understand it, and then we all add up. I realize that when I'm talking plenty, you tend not to be hearing me. So I want it to come from you. Then I'll add up. Yes, Zuera. Today you are talking, pa.
Okay, sir, please, I want to add up to the historical aspects. No, formerly, they were part of the kingdom plantae or the plant kingdom. However, because they lack chlorophyll, they have been taken out. And they were taken out because they have some unique features in them. That is why they were separated from the kingdom plantae. Then also they are eukaryotic organisms, meaning that they have memory-bound nucleus and organelles, and then their nucleus are well defined, unlike that of the bacteria. Thank you.
All right, thank you. Who else want to add up? Yes, Rebecca.
Sir, please, I just spoke. I'm Rebecca.
Oh, okay. So Zuera didn't talk. All right. Hey, I'm coming to call the name now. The name has disappeared. Zuera, yes, I said you can talk. You are not talking. Your hand is still up. Okay, let's take Alice.
So from what we read, fungus is within the kingdom fungi, and it is said that yeast, mildews, mold, and all that. That in the basic school, our home economics books, then when this microorganisms in terms of food spoilage, fungi is mentioned, mold, and then these things that are here, we mentioned them. How are they related? Are they the same thing? Because I don't get it. If they are off, then why is it that we list them separately? Yeast, mold, then the fungi itself as agents of food spoilage.
So the answer is simple. The answer is collective or most microorganism. The fungi, okay. Fungi is actually having yeast and mold under it, like it is part of yeast and mold are under fungi. Now, the whole thing is that you see, just like we're talking about the genus and then the species and then the subspecies, it come in here. But at that level, you cannot go and tell the child that fungi, yeast and mold are all fungi. Like the child will find it difficult to comprehend. But from what we are learning now, I should tell you that some things can be grouped under the same umbrella at a particular point in time. So, for instance, when you are talking about food spoilage, okay, we have something called the mycotoxins. Mycotoxins are fungi that are formed from other toxins, which are known as the aflatoxins. And then the aflatoxins are also having another group under them. So normally we say that these aflatoxins, what they do is that they affect our cereals. And when they affect our cereals, it makes it bad for us to eat, because if you don't know you eat it, it can cause severe kidney and a liver problem. So fungi infection, which is particularly mold, and then the mold we are talking about here is an aflatoxin. So fungi, and then under the fungi we have the mold, and then the mold we are talking about is what? The aflatoxin, and it causes food spoilage in cereals. You get it? So it is very much related, but then at that level, you cannot go and tell the child that yeast and mold and this thing are all fungi. For instance, after saying that, then you go and say mushroom is also a fungus or a fungi. How then do you think the child is going to comprehend at that level?
All right, so I get it.
You see, so that is why maybe in those books they brought it down, or they have to streamline it for you to. But then that notwithstanding, they are all under the same group. Any other question? Oh, Charlie on. This. Yes, any other question?
Sir, please, a lot of hands are up.
Lydia's hand is up. Lydia.
Okay, Lydia, please talk.
Oh, sir, sorry, please. It's not a question, but I just want to know if we'll go for a break.
Yes, we'll go for a break right now. Who else? Yes, Laurie.
Sir, please, I'm asking a question.
Okay.
Sir, please, the eukaryotic organisms, please, can you explain? Thank you.
When you say an organism is a eukaryotic organism, what does it mean? Yes, Esinam, Bodo.
So, my understanding of a eukaryotic organism is that it means it has a definite shape. It means that the thin layer that surrounds the cell, that is the membrane, giving it a definite shape.
Okay.
That is my understanding.
Okay.
And then normally, animal cells are prokaryotic. But in this case, we have specific animals or some unique animals that also have a definite shape. That is the cat and then the dog we are talking about.
Okay.
It means they are unique. Animal cells are normally they normally lack membrane-bound
organelles.
Organelles, but in this case, they are not lacking it. So it means they have a definite shape. That is why they are classified under the eukaryotic cell. Likewise, the apple plant, because generally plant cells are how do you say it? Eukaryotic. It means they have a definite shape. But it means apple trees are also unique in the kingdom plantae. And so that is my small explanation to it. Thank you.
Yes, Eunice.
Okay, so I also understand it that there are cells that contains nucleus.
Okay.
Yes, and other bounded organelles.
So they have a memory bound nucleus.
Yes.
Put it like that.
Yeah, okay. So they have a memory bound nucleus. So when you take the animal cell, the plant cell, and other fungi, they have memory. So it doesn't mean you can't say some plant cells have are eukaryotic, or you just check the membrane or the nucleus. If they are membrane bounded, that means that organism is eukaryotic. Thank you.
Okay, so let me just add up. Please, the person who asked the question, we are saying they are eukaryotic cell because they have a memory bound nucleus. They are also multicellular, and then they also have a memory bound organelles. You get it? So directly or indirectly, they are just, it is directly opposite the prokaryotic cell, where we say that it doesn't have a memory bound nucleus, and then its internal structures are not also memory bound. You get it? So when it comes to the eukaryotic cells, their internal structures are memory bound. They have a discrete shape and all that. Is that okay?
Yes, sir. Thank you.
All right. Eunice, your hand is still up. You are done answering a question. Do you want to ask a question?
No, it's a mistake.
Okay. So it says that whilst mushroom and toadstools, into bracket, poisonous mushroom. So the toadstools are the poisonous mushrooms, are by no means the most numerous of or economically significant fungi. They are the most easily recognized ones. So what it's trying to say is that out of all these fungi we have mentioned, the most easily recognized ones are this mushroom, that is the one we eat, and then the poisonous one are the most commonly recognized fungi that we normally mention. So fungi other than these mushrooms are sometimes collectively called molds. So this comes in to answer your question, Madam Alice. Madam Alice, are you here?
Yes, sir.
So it says that fungi that are not mushrooms are sometimes collectively called, all the group of those fungi are collectively called what? Molds. And it says that although this term is better restricted to fungi of the sort represented by the brand mold. So you see, the mold is a collective of fungi which are not mushrooms. You understand? So all we are trying to say is that fungi, they are unicellular. They are unicellular, and then they include yeast mold and other groups. We have mushrooms under them. Some animals they are eukaryotic in nature. We have the dog, the cat, and all those things. So talking about eukaryotic cell organisms, what are they? They are organisms that have a discrete memory bound, and then their organelles are also memory bound. They have they are multicellular. They are multicellular, which means they have more than one cell. and they have a clearly defined nuclei. So their nucleus, because it is memory bound, it is well defined. So those are some of the things you say about fungi. And then I want to know if you have any question or not. Any question? Any question? Hello?
Sir. Hello, sir.
Yes, Rebecca.
Sir, please, does it mean that collectively, if we should put yeast, rust, smalt, and mildew collectively, they are called molds? Thank you.
Yeast is a different thing. So it says that statement there is not how do you call it? That statement there, I want to use a word that it's not generalizing, okay? That statement there is not general. You see, sometimes we have a lot of things under one that you cannot just be mentioning everything. But yeast itself is a different thing on its own. The mildew on its own is a different thing on its own. You get it? Mushroom and all those. So the ones that they have mentioned are all different things on their own. But if you go into the molds themselves, we have a lot. We have the mycotoxins, where I was talking about the aflatoxins, the endotoxins, and all those things. Like most of them will be there. We are just being introduced to the basis. We didn't go much into it. You'll be doing microbiology, where you go into food microbiology, where you go deep into all these things. You get it? So I don't want to take you down there plenty. Is that okay?
Yes, please. Thank you.
Yes, Mabna.
Sir, please, my question is, how do you determine that this is poisonous mushroom and this is not a poisonous mushroom? How do you see that?
If you're asking me this question, me, I'll say I take it to the lab. But those who are there, those who farm, they know it. Are you and I don't farm mushroom. I don't even eat mushroom. I've not eaten some before, so I can't tell.
So please, the ones we don't eat, we consider those ones as poisonous mushrooms.
Well, I can't really tell. So if I want to know what is a good mushroom and what is not, I will tell you that I take it to the lab. But those who farm, they know it. Yes, Alice.
So what she asked, I think we have different types of mushrooms, and what we here eat. Some people somewhere don't eat that because probably they don't know that it is edible. Country that they showed some poisonous mushrooms, and they are very huge. They are huge in size, and then they are very colorful. Some are brown with spots like red. They are very, very colorful. So to me, I think the color of the mushroom can even determine whether it is edible or not.
Okay.
Sir, please, and if I'm to add to what she said, every poisonous plant or tree that grows mushrooms is also termed as poisonous. That's what I know.
Okay. So we are taking it from your colleagues for the mushroom. I don't have much experience about mushrooms, so let's take it from your friends. Yes.
Hello.
Alice. Grace.
Sir.
Yes.
My colleague said the color will determine. Please, it's not always the color, because we have some mushroom that grows on a plant, like the isan. The mushroom that grows there, they are colorful. They are some like gray color, just like they looks like the one that grows on palm tree. But this one, they are rather deep gray. So if you use the color, you might get it wrong. That's what I want to add.
Okay. Please, Have you heard it? Me, I have no experience in mushroom. That's why I'm not talking about mushroom. But don't worry, I'll search for some and then let you know, okay? So for instance, when you check the slide, it says that the toadstool is a poisonous mushroom. Toadstool. It's giving you an example of the poisonous one. You can see it here, right? But I don't know how it actually looks like. That's why I don't want to talk, please. So that is it. But I'll get examples of them and then give it to you if you want to know. Maybe you really want to find the differences. That'll be fine. So let's break for about 20 minutes and then come back so that we'll come and do the lecture 5. But before then, do you have any question again? Hello?
Hello, sir.
Yes.
Hello. Sir, please, please, are you Ibrahim? Say Baba.
Am I what?
Please, we were given an assignment, but the name, I wrote Ibrahim Say Baba. That is integrated science lecture 2. Please, the integrated, is it the same as lecture 1 or 2? I don't understand. So I'm doing the assignment and I don't know where to. They say we should do it PDF to email. But I don't know his number.
I'm much confused. I don't really know what you are talking about.
Please, I would like to know if you are the Ibrahim Baba Said.
Ibrahim what?
No, he.
Hello?
Mr. Israel, not Ibrahim.
Yes, he's not Ibrahim, please.
You have joined a different platform. So you check again because he is Mr. Israel.
Okay. I think last 2 weeks we had a lecture called this week, and that's why I'm not getting a different group. This is a group for all of us, so I'm on the right week.
And he's not Mr. Ibrahim, he's Mr. Israel. Maybe Mr. Ibrahim is another center, not this.
Okay, please. I don't think I have actually given any assignment. The only thing I gave is some short assignments. Listen, wait, switch first here. The only assignment I gave is the one on the course outline, the one that we say that you should write not more than 2 pages. The benefit of soap, detergents, and all those things to your field of study. That's the only assignment I've given. Even I have not told you when I'm taking it. So please, I've not given any assignments. Okay. So let's break for some 20 minutes and then come back. So come and do the lecture 5. Mr. Ima.
Yes, boss.
Please, I hope you received the things I sent to you last week.
The 3 slides.
No, I think I sent some sample questions as well.
Yes, we received the sample question in addition.
Okay. So those that you couldn't understand because we have not done the lectures, as we are doing it now, I guess you are getting answers to them.
Yes, boss.
Or the answers were there. Were the answers part of the questions?
Actually, I forwarded it, but I myself have not gone through it.
Okay, because if the answers are part, then I did a mistake because I should have given it to you for you to solve and get the answers.
I think it will be part.
Ah.
And not part 2.
Yeah, no, the answers are not parts.
Yeah, that's what I want to do. So it means that I've done what I wanted to do.
Hey, now, I know you.
No, the answers are not fact. So think that you have not opened it. Some of us have opened it.
The way you came in fast, you know.
Yes, I need to come in fast so that you will not mislead anybody.
But
So sir, the answers are not bad, but as we are doing the lecture 4, we are also getting the answers from some of the questions.
Yeah.
Okay.
So I actually gave it to you because it will guide you as we are learning. You know, when I'm talking about something, you know how the question is going to be asked and those things.
Yes.
So it will guide you. So try as we learn. You try to solve is always try to be solving until you are able to solve all the questions and get correct answers for all of them.
But say you can give more, or that's all.
Hello, sir. Some of us don't have it too.
Sir, do you have more?
Hello, sir. Some of us don't have it too.
Unless I
it's on the page.
To set more.
Which page?
Yes.
Question or different question?
Please. It seems
Hello.
some people are from different platforms and they've joined us and they are drawing us backwards, and certain things that we've already shared, they are still demanding for it.
Please go
Not
for
that
break.
it's really bad. Okay, so we'll go for break. When are we returning?
20 minutes time.
Just
20 minutes time.
20 minutes.
20 minutes.
Yeah.
Okay, then we'll come at 10:10.
Yeah.
Hello.
10:10.
Thank you.
So as I was saying,
What is
not
your
that
time,
it's really
No, Sayima, please, we come at 10:15.
10:15 rather.
Yes.
Yes.
Oh, okay. 10:15. So as I was saying, this platform is open and anybody can join, but it is open for specific centers. So I will plead those who are not part of the centers that are supposed to be here, when you come on board, yours is to listen, not to ask questions or not to ask things that does not relate to us. Because if you ask certain questions that is not relating to us, it will confuse all of us and it will draw us backwards. Meanwhile, you have your own platform or your own center to study. So I will plead, if you need something, you can ask backdoor or you can ask on the chat form or after we finish everything. Because our tutor has not given us assignment, but we come and raise that he has given us assignment. We are already occupied and we are stressed up. So I will plead with you. I will plead with you. If you are not part of us, when you come, you are coming to let me say
Observe
as an yes. And to be and so I beg of you. This is a humble plea.
Hello.
But it's not
and she was saying the platform is all for all of us, but there are some send you.
It is for specific centers.
All right, but all can I say hi now center? What's the center is for all of us?
We are all one. What I'm trying to say
it's
is
good
we
you
are
have
all
seen
one.
that she's lost. So let's move on. Hello, Brahima. Hello.
Yes, we are all one. That for foundation and curriculum, that platform is specifically for Ganas and Sunyani. But we have a lot of centers that are also on it. And they are on it only to observe. You can't prevent anybody because it belongs to all of us. But they are there to observe. Like the way we were given the assignment, a lot of people were calling me. And those people that were calling, a lot of them, they are not part of the center, so they are not part of the assignment. But some are doing it they want to present it, but you don't fall under the tutor center. So if you see a question, it is for you to study, read and learn and study. I can go and join another platform, but I'm joining only to get what is going on and also to study, but not to engage in it. So I will plead, those of us who are on this platform, it's not all of us that are supposed to be here. That is the truth. There are some who are going face to face, but they are not able to go to the face to face, and they try to join online for the same thing. It is very good for you to do that. But when you join, just listen, not to engage, because you know you are not part of it. So that it will not cause another thing for us. Because we know we have only 3 assignments. We have Foundation 2, we have Curriculum 1. Even nutrition and health. We don't have any assignment there. But others are calling, have we've been given assignment? And I said, we've not been given any assignment.
Hello.
Yes.
Please, do you know something? Let me just come in. Not to offend anyone, okay?
Okay.
You know, sometimes your colleagues at the other centers are not enjoying what you are enjoying. So, in a way, when they join you, they feel bad, okay? So, I think at the end of the day, you are all doing the same course, and then the most important thing is for everybody to be able to pass the course and pass well.
Yes.
So, what I would wish you do is that maybe when they come in like that and we see that you have a nice way of, addressing them or sharing information with them. I feel nothing bad with just that sometimes, you know, when people have information from outside, they begin to see their tutors or other people, they say things that though the person is just trying to appreciate what he or she has had outside their jurisdiction. But at the end of the day, how we normally say it as students, make it feel offended because the person at the other side would think that, oh, so my students are seeing me not to be doing the right thing, and then they see other persons. So that's why normally I tell you that don't share anything I give out with people. But that notwithstanding, I feel like so far as we are all doing the same course, you write the same exams at the end of the day. So among yourselves as students, you can be of help to each other. Sometimes you can just compare, share ideas from. So
and I've been doing that. I've been having calls from different angles, and I respond to everybody because we are all, at the end of the day, we all need to pass and go because we are in this together. It will not be good if somebody gets a refer. That one, dear, I'm not praying for that.
Yeah. So just have a nice way of addressing yourselves.
I will do that. Very good, very good. So I will still engage them and then talk to them. Because I have a lot of colleagues who are going face to face in Kumasi and UCC. I still share things with them because we are all one. We all need to go one. But those who also join that needs to share. You share or you come for it quietly. But if you engage that, the owners themselves too will also find themselves wanting. That is where another issue also comes in. So if I've made any statement that goes against such people, I will plead for that, for them to forgive, so that we move together. And then
hello, Seima.
Yes.
Hello. Please, Riverview. So who will be our tutor?
The thing is on the timetable, the link, the names
because
of the tutors.
Mr. Israel have been teaching us since.
Yes, if Riverview is Mr. Israel, it is there. It's about 5.
I heard you saying Ghana's and
that's why I've not mentioned any center.
That's what you said. That's why
I mentioned Ghana's.
Yes, that's why I'm a little bit confused. I wanted to
said
verify.
other centers. I did not make. I was not specific.
He was referring to that of the foundation group.
That foundation group. Yes, and then the foundation group. I'm talking about the foundation group.
Okay. Then we did not have the questions. We don't have that questions.
And so you WhatsApp me backstage. I will give anything I have with you.
Okay, if you wouldn't, can you please put it on
and the thing is, I
the
beg
refs
of you,
platform so that
our group, this group is made up of almost 100 participants. You get it? And these 100 participants, each and every day I get calls. I send documents to people. People ask of it one after the other. I can at least at times I getting to 20 people who have misplaced their slides and things, and then they are calling me for new ones. You see, that makes me to compile all the slides for the subjects and then put it on the platform for you see our for science we have Only Child, we have Zenith, we have Riverview, and then we have Sunyani. Is these four?
We also have
this,
fine.
No,
hello.
Listen, please listen. UCC, Wesco, Ganas, Mauli, they are also having their platform. These four centers that I mentioned, they are having this platform.
Please, Serious is among, just that they didn't write it on the timetable.
Oh, okay. I'm looking at the link that they sent.
Yeah,
it has only
it was
4.
on the audio.
So they could be part of it. So those that are part of it, they are all here. That's why we are all one, as I said. So please, we are all one. We are all running towards one goal. So let's move.
My problem is when you put it on the refs platform, then the refs will push it to their own platform. So that's what I'm saying. So if you wouldn't mind, you can send it to us so that we push it to our friends on our platform. We have a
Wait, what are you talking about?
the science questions and
Please, what?
Hello.
The science is on the science platform.
I will put it on it again.
Okay.
It is on the science platform. I'll put it back.
Yes, it's there.
Okay.
I'll put it back on it right now. Okay.
Okay. Thank you.
No.
Hello, Brahima. Hello, Brahima.
Okay. Yes, madam.
Hello. Please, I'm not on the platform. Can you please add me to the platform?
What center are you?
Zenith.
Brahima. You will put you there.
Please, who says he's at Zenith?
Please, is that what?
The one who said he's at Zenith and her name is not on the platform.
Millicent.
Please, your number.
Okay, 0550.
Hello, please.
0550.
Did you ask if she is a rep or what?
I'm not a
you,
rep.
you have to verify. So she can't be on that platform.
Okay.
Please, where
can I get the slides? Okay, please, where can I get the slides?
That's why we want him to put it on the refs platform so that we share it.
Call out your number so that she will add you to the group.
Okay.
Madam, please, your number. The one who says it's at Zenith.
Okay, 0550 25 98 17. Please, should I mention it again?
259817.
Yes, please.
Okay.
Thank you.
What course are you offering? Is it clothing or food and nutrition?
Clothing, please.
Okay.
Okay, thank you.
Hello, members.
Yeah, hello.
Hello.
Brahima.
Yeah, please say.
Hello, Brahima.
Hello?
Yeah, please. You have to. You
It's on
should
break.
help me to them.
Please, some of us, we need like the way we should go to YouTube. The way you can go to YouTube and get the recording back.
Hello.
Brian,
Yes, I'm listening.
please, can you please educate us how to go to YouTube and get the videos to watch them?
Oh, okay. But they've put some of the
Hello?
videos on
No.
our. They've put
On
some
the
of the
YouTube
videos on our yes, on our platform,
and
the
then
main one.
so when you tap the course code, they will give you a different thing altogether.
That is another thing altogether.
The guideline.
So me normally I try to listen to this one rather than going for the record because this one is interactive. You're able to get everything.
It's EVT. Please, are you listening? Please, are you listening? The one
I'm
on the
listening.
YouTube.
Please stop.
Brahima, the issue is that I learned that you can go to YouTube and just to go.
Your line is breaking.
Or it's only recording.
And your line is breaking, so we didn't get you.
I said,
Uh-huh.
can we go to YouTube to get some of the videos that work, or it's only a recording alone?
And it's only the recording. You won't be able to get a video because we are not taking a video. It's recording.
Okay.
It's recording.
Hello.
I'm putting the trial questions back on the science platform. Those who are not on the science platform, please exercise some patience. I will put after the lesson, just give me hi and I'll add it to it. And then you'll be able to get everything. If one thing too, I beg of you, there are certain at times. Please, are you listening? Please, are you listening? I beg of you. At times you may mix or you may lost some of your slides or documents. If you try to ask from me personally, it means that if about 80 people demand for it, I have to test 80 things, the same thing 80 times to everybody. And that one will put a stress on me. So I beg of you, if you lost any document, just ask and I'll put it on the main platform so that it will be only once a week, and then you will get it. Whilst others too who may also be experiencing certain the same scenario of mixing or losing the document, they can also get it so that only one person will ask and I will put it there. Anyone who have need it will go for it. But if you ask me to send it to you alone, tomorrow another person will also ask, I will send it to that person alone. Tomorrow another person to ask. So just ask and I will put it on the major platform for all of us. Please, do you get me?
Yes, please.
So that is what I want to say. Those who are not on the science platform, you just give me and I'll add it to it. Then off we go.
So is our science lectures over?
No, it's not over. We have just a minute to continue.
I'm over. The network didn't favor.
Hello. Hello.
Yes, Lydia.
Yeah, please kindly add me to the reps platform, okay?
Please, some of the rest, we are pleading you. We are pleading you. When you say you want to be the rep, do the work, please. Some of you call, you not even mind us.
We have all the names and their contact number as reps, and we form another. Please, I'm coming. Please, I'm coming. And we form another group. Please, I'm coming. And we form another group of all the reps. So we have about almost 10 centers as rep, and we share things over there together. Should be given to each and every center that needs an information. That is what we are doing. So if that person's name is not the right person for the rep for that center, let all those who are there just come together, get one person that can be the rep, and then you send it to me. I'll remove the person's name there and I will put that one that you provide. If it is the same person that is supposed to be there that is there, then that person must also respond to the people that are in the center with him or her.
And Brahima, do you know that because they don't respond to our calls, that's what we are calling you to worry you.
Oh, me, I don't see it as a worry. I don't see it as a worry. It's part of me.
I want to get it clear.
Please, our time is up.
Hello, no,
our time is up. Yes.
Say this issue about the reps thing. No, the reps we are not paying them. So let's not make it look like they are under any command to answer our calls or respond to us. Some of us, the way we act, is even also annoying. Sometimes somebody, even you yourself. Wait, please let me land. Sometimes you post something on the page, somebody will come and will not even go through the page to see what is there, and will come back and ask the same thing, or even call you.
That one there is true, but I've experienced more than 20 calls on this.
Please.
Is there anybody at Zenith that have not answered before?
Oh, Zenith, you are doing well. Florence is always
so. I
point.
beg of you. Let's work.
So please, if you are here and you are in Zenith, see Florence. Florence, she's on the platform now.
Please, our time is up. Let's continue with this discussion after the lesson, and it will really help us to iron all differences and then help. We as reps, we are facing a lot of challenges, and then you too, you are facing challenges, but we help each other. And just like one of the reps said, the thing is there, but the person will not scroll to go up and get it. We will rather wants to ask you. And I'm telling you, to be frank, some of the things, if I want to respond normally, it will create problem. So normally at times I don't respond to certain questions. I'll only go straight, then give the thing to the person, and then we are okay and we are going. But I have every right to tell the person, go and scroll to the top and get the document you want. It is there. You know, the questions of foundation, we put the question there, and just about 3 or 4 slides upwards, the question is there, but somebody is asking, where is the question? Personal, sending me message personal, where is the question? Where is the question? And the question is there. Just about 2-3 slides upwards. So for the reps, we have them with us, and they are all part of us. Let's exercise patience for them. Let's come together. Let's do it together, and then we'll be okay, all of us. Thank you very much. Let's continue with our lesson. Mr. Israel, please. Our time is up.
Say, my name is Onisu.
Also, ebia or boabo and yaman one and say, wa peja ni mukakra, but also our online.
Yes, sir. As we doing.
Yanga also, yeah, sawa how a lot of faith over here. So, and yes, I know I discriminating.
Some may hear research on it, so I have to be greeting you every day.
Oh, Buakura, oh, research, baby, how I do no first see or miss it out.
So, catch me as I can make your research.
Florence, miss it out first see.
Say, Mammy, and to this, so. But when they
Florence,
one day, come back to science.
Florence, miss it out. Why you may know the idea, Yanko?
You see, Monday, yeah, science.
Hey, Monday, our will for us, Sunyani and Ganas. Hey, is it Sunyani and Ganas? We have food and nutrition. Nutrients.
No, but no, Madam
nutrition
said that she will take us on a Sunday.
No, she's not part of our us.
Yeah. Sunday. He said we'll
yes, he said that now she's free that she can
from
take
11 to 4.
Yes.
So 11 to 3 or 4. We'll have it.
Okay, yeah. So next week, according to the timetable, is practicum, right?
Yes, but there was an announcement that it will be a normal lesson. For my investigation and what I asked, it will be a normal lesson to complete our lesson so that we can have our quiz next week Friday. This coming Friday's week.
Is it Friday?
Hello, it's 21st. 21st is Friday, or?
But they told us we will be coming to the center to do the quiz.
No, quiz is online.
They said
Madam,
one
quiz
face and one
the
is
first quiz is online. So they say we'll be using a software called Kiosk. It's even on the platform.
No, she said that
you if you don't know how to use it, if they say if you don't know how to manipulate it or use it, then you have to get to the center. There is somebody there to teach you.
What we were told during the orientation is that with the first quiz, we all have to be at the center.
Good.
It is the second quiz that you have to be at your various destinies.
So
the first quiz, you come to the center, and then a code will be given
with your
to
devices,
you.
without devices.
And then we will be having invigilators as well.
Okay, but okay. I will still inquire more.
Okay.
He said, you see, some of the things they've written the documents, that is the timetable, the link, and they've put centers on it that are supposed to go online and the tutor that is supposed to take them. But during the audio that the coordinator gave to us, I think 2 days or a day before the beginning of the course, she added other centers that will also be going online, which was not on the timetable or the link table.
Exactly.
So you don't get how the thing is. So if you don't inquire on certain stage, you'll be taking old direction and later it will backfire. So we need to
She was the same coordinator who explained it to us. Do you know that on the first day that we were supposed to do online, some of us went to the center.
Yes, some went to the center.
Yes, so she was the same coordinator who explained. In fact, she had time to explain things to us. And then she said that for the first quiz, we will be coming to the center there with our devices to do the first quiz. Whilst the second quiz, we'll sit in our comfort in our home and then do it.
Okay.
Went to the orientation. That was what they told us.
Yes.
Okay. Then that would be very good. Has been the first time of doing that. If we are all together and then we are being directed,
I'm sure probably they want us to when we come together and then we know how to go by so that the second quiz, when you are in your comfort home, it will be easy for you to how to operate it.
Yes, I think so.
No, please. What I know is that the first quiz, we are going to be seated with invigilators invigilating us.
Yeah, that's what I said.
You are not just
Without
going to be,
devices, we are not going to use pen or
with your
paper to do it.
devices.
Yes, that's what I said.
So there are rules. You cannot just
Yes.
come and then be doing anything. You'll be
But can
someone
we take our calculator?
watching you.
Yes, we
Can
have
we take
it.
our
Yes.
calculator then?
Oh, you send everything. In your exams room, things that are allowed, you send it along. Things that are not allowed, you don't send it along.
Hey, when are we
It's always
writing
allowed.
the quiz? When are we writing the quiz?
22nd, 20.
I think 21st.
22nd and 23rd. Next 2
Okay,
weeks.
22nd and 20. Next 2 weeks from now.
Yes.
If you say next 2 weeks, you are entering into 3 weeks. 2 weeks.
And
from
then,
now.
and then I want
2
to.
weeks. 2 weeks from now.
Hey, Sir Dennis.
Miss,
So please, the next week practicals, what is it about?
hey, I said we
Yeah,
are
thanks,
candidates.
yeah, I
Yeah.
want it.
Oh, I was
I
off
said
for
we
some
are
time.
candidates. Yeah. Okay, let me repeat for you. Through inquiries, they said it is there, but we'll still use it for lessons. So I will ask again and come and confirm to you again within this week before the weekends.
Okay.
Okay. So Bosu, with the issue of the practicals, what we were told the other day by the coordinator is that they place it on the timetable.
Okay.
So if within that particular block you are having a practical course, that's the day that you are going to use in doing that practicals. But if you are not having any practicals, then of course it will be the normal lecture. Yeah.
Your practical. Okay, so please let's welcome the
All right,
teacher
so
to
please
continue.
you let me finish and then you continue with your discussion, okay?
Okay, boss.
All right, so we are moving on to look at microbial activities in food and textile, okay? So what we are going to be talking about in this lecture is we are going to look at, first of all, how microbes grow. So the outline says that we'll explain factors that influence microbial growth and development on food and in textiles, then describe the effect of microbial activities in food and in textiles. Describe how microbial growth and development in food and textile can be prevented, and then discuss the benefit of microorganisms in food. So these are the things we are coming to look at for this very class. I would be going a little bit faster if it wouldn't be of a problem, because most of the things are just, they are quite simple to understand. I'll give you the general idea, and then you would, as you read, you try to get examples, more examples for yourself, so that if I'm able to finish this within 30 minutes, next week I'm not supposed to meet you, but then I'll try and meet you for the last lecture. I hope that is okay with everyone.
Yes, sir.
Yes, I was.
Okay, so I'm trying my best to do this. If I don't even finish, I should go far, so that maybe if I'm meeting you next week, I'll just do this and then finish the other one with you. So when you talk about factors responsible for microbial growth, you are looking at certain factors that allow for microbial growth. And I think when I was explaining the previous point, I mentioned some of them, okay? But now we're actually coming to look at them one after the other and how they influence the growth of microorganisms. So we said that microbial growth have 4 phases. We have the lag phase, we have the log phase, and then we have stationary phase. The last phase is the death phase. Now, at the lag phase, that is the phase at which the microorganism becomes present or is present. So it says that the period where the individual microbe is maturing or not yet able to divide, synthesis of RNA, enzyme, and other molecule. Okay, so here, for example, if I have exposed my food to an environment, the microorganism becomes present, but at the point it becomes present, at the exact point it becomes present or is set on the food or on the clothing, we call that phase the lag phase. Okay, the lag phase. At that phase, the microorganism is present, but it is not actually multiplying. It is now maturing. It is now going to mature. And then as it matures, it gets to a point in time, as I told you from the beginning, that microorganisms, when they grow, they multiply. They don't increase in size, okay? So this microorganism will mature to a point in time, and then it's to start to divide. When it divides, the division will continue based on certain factors or certain consideration. So if those factors are available, the microorganism will continue to increase or divide in larger numbers or quantities. As this increase or division continue, it will get to a point in time that the division will be stopping a little or it will be coming down. And we call that phase the stationary phase. So you realize that at the lag phase, the microorganism became present. At the log, we have lag and log. That is L gene, and then we have the LA gene, and then the LO gene. The LAG phase is where the microorganism become present and start to mature, but that place is not yet dividing. At the log phase, now it matures to a point and then the division starts. So here there is an exponential increase in the number of microorganisms, or in the number of, we call it colonies, in the number of colonies present on the surface or in the item. Then as these colonies continue to increase, you know that they are using certain factors such as nutrients. So you realize that when the nutrients that is there, they are using the nutrients, it will get to a point in time that the nutrient will be what? Depleting, or it will be what? It will be getting finished. So as the nutrient gets finished, you realize that now it becomes the survival of the fittest. So if you are strong, you are able to survive because you'll be able to get more nutrients to survive. So we call that phase the stationary phase. And we say this is the phase at which the number of dying, there is an equilibrium, or there is a balance between the number of dying organisms and then the number of what? Surviving colony. So number of dying colony and then surviving colony becomes equal. When that exists or that happens, we say that that is a stationary phase. There's an equilibrium between dying colonies and then surviving colonies. Then we have the last phase, which is the death phase, where there is a decline. Okay, there is death. So now they die plenty. The death becomes more. That is the 4 phases of microbial growth. Is it understood? Yes, please.
Sir, I want to understand something. So with what we just learned, the lag stage, the log stage, and all that, in normal terms, can we say is the lifespan of the microbe?
So the life cycle, let's say the life cycle.
And please, enzymes. I want to understand enzymes. Are they also microbes, or they are different thing altogether? I don't get the relationship between enzymes.
Enzymes are not microorganisms. Okay. Enzymes, we say that they are biological catalysts. They are biological catalysts, okay? And their role is that they are produced to speed up chemical reaction within a substance or within a surrounding. They fasten or they speed up the chemical reaction without they themselves being what? Consumed. So enzymes are not microorganisms.
Oh, okay, so thank you.
All right, Grace.
Example of microorganisms.
I should give you an example of microorganisms.
Eh?
I
want to know.
Hey.
Bacteria is an example of a microorganism.
Okay.
Bacteria.
Okay.
Yes,
They are simply organisms that cannot be seen with the naked eye. So any organism you can't see with your bare eye unless you use a microscope is classified as a microorganism or microscopic organism. They are very tiny, minute. Thank you.
But I'm speechless. I'm surprised because
Say
Hey. No. Like this is a scary question. Mr. Emma, what do you think?
Oh, sir, I was lost.
I only say
Say no comment. Say no comment.
No, like, do you know what I guess for? I felt like I have poured about 1 barrel of water on stone.
Oh,
That's a man now, but three.
Everyone is normal different.
Well, it is well.
It shall be well.
It is.
Hey. When we went for the break, she went and eat banku. That's why.
Say, you know what? My mind was on amoeba and agar and the rest. Forgotten what we just discussed. That's why I wanted to be sure.
Okay, somebody says it is well. It happens. Okay. Let me just take it like that. Okay, so as she said, microorganisms are minute organisms you can't see with the naked eye unless we are aided by a microscope. And we have been mentioning examples of microorganisms here. We mentioned bacteria, we mentioned fungi, we mentioned mold, yeast, we mentioned viruses. All those things are microorganisms. Please, is that okay?
Yes, sir.
All right, thank you very much. So let's look at the factors that influence microbial growth in food. Okay. Now, when we talk about the factors that influence microbial growth, both in food and in clothing, these factors are grouped into two or can be grouped into two. We have the internal factors and then we have the external factors. So the internal factors are referred to as the intrinsic factors. Internal factors are referred to as the intrinsic factors. And then the external factors are referred to as the extrinsic factors. Under the internal factors, we have factors such as nutrient content, the pH and organic acid, water activity, and then the biological structure of the food. Then when you move to, and for the clothing people, it will be the same thing, okay? So the nutrient composition of the the fiber, the pH, and then the organic acid of the fiber, the water activity, and then the biological structure of the fiber will all come to play. Then the external factors, we have relative humidity, we have temperature, and then we have the gaseous or gaseous environment, however you want to mention it. Yes, Bodo, your hand is up.
Okay, I took it off anyway. I don't know whether I'm sending you back. I think her question was example of enzyme. If I heard her correct.
No, she said examples of microorganisms.
Oh, okay.
Madam Grace, please, were you looking for examples of enzymes or examples of microorganisms?
So I asked for examples of microorganisms. Thinking amoeba and the rest, not knowing it's right here.
Okay, then I'm sorry.
Sure. Right. Thank you. Okay. So as I was saying, we have the internal factors and then the external factors. So now we are going to pick these factors and look at them one after the other. Okay. Now we are taking nutrients. When you talk about nutrients, you know that human beings or living organisms, we can only survive when there is nutrients, right? That is why even plants, they make food for their own consumptions, for them to be able to survive. The plants make their own nutrients for them to be able to survive. So, for instance, we are saying that nutrient content of the food. Now, the nutrient composition of the food can influence the growth of microorganisms within the food or within the environment. So if I have a food which is rich in protein, carbohydrates, fat, and oil, and other nutrients, it makes it possible for microorganisms to be able to grow. Just like human beings, if you don't eat, you don't have nutrients, and for that matter, you are not able to grow or nourish, right? Hello? So same applies to the microorganism. Now, let me ask you this question. Take, Alice, I'm coming, okay? I'll let you respond. I'll call you later. Take meat, okay? Meat and cassava. Leave all of them at room temperature for 24 hours. Which one do you think will get spoiled easily or faster, and why?
The meat. Say the meat.
Why? Why is it so? Yes, Grace.
So I say the meat because of its composition, the nutrient content of the meat, and the kind of biological structure of the meat will cause the meat to decay earlier than the cassava.
Okay. Who else has a different thing to add? Yes, whose hand is up again? Rebecca.
Sir, please, I think the meat is exposed. It doesn't have anything covering it. It doesn't have any protective kind of thing. But that of the cassava, at least it has something covering the actual part that is to be eaten, unlike the meat, which is exposed. Thank you.
But what if the cassava is also peeled?
And that is where the issue will be.
Yes, Elizabeth, you also want to contribute?
Sir, please, I also think that protein content in food causes food to spoil earlier than the carbohydrate content in food. That's what I think.
Can you reconstruct the statement? You are making a point, but I want to reconstruct it.
Protein foods spoil earlier than carbohydrate foods.
Okay, I think I'll think. Yeah. Kodo, you also want to say something?
Yes, please, the availability of water or the water activity in the meat is higher than in the cassava. And so it will create the room for the microbes to multiply.
That is also true, but I'm actually focusing on the nutrient alone. So let me take what she said is correct. She's right, I just want to take more views.
Okay.
Vivian.
Listen.
Okay.
Me, I also said that the microorganisms found in the meat are higher than the microorganisms found in the cassava.
How do you know that? How are you able to detect that?
Because meat, let's say meat is a protein food.
Yes. It's okay. Okay, Mr. Emmanuel.
Okay, so I also say that the meat will spoil easily because of the protein and then the water content in it. This is because bacteria, they strive more, or they grow or multiply more where there is moisture, humidity. So for the cassava, it also contains water, but the structure of the cassava and the structure of the meat, in terms of the protein in the meat, gives it more room for it to spoil easily than the cassava. Thank you.
All right, thank you. That's very nice. So we are all we are trying to say is pointing at one thing. Protein. The meat has a lot of protein, more than the cassava. That doesn't mean cassava doesn't have protein. Okay, cassava has.
Vivian, please mute.
I know some of you will disagree with me, but then cassava also has protein. Is that okay? Likewise, meat also have some form of carbohydrate or some amount of carbohydrate. But then when you take the protein in meat, it's very high. And for that matter, just like the way human beings, when you eat a lot of protein, you grow very well. Same way, microorganisms also feed on this protein for their growth and their multiplication. So because meat is rich in protein than cassava, definitely meat will go bad or will get spoiled faster than what? Cassava. Is that okay?
Okay, sir.
So
yes, sir.
We are all right.
We are all right.
Then,
Hello, sir.
Yes.
Yes. Sir, please, my hand is up.
Okay, talk.
Okay, talk. So, please, if I may get it right, so that does it mean that what do you call it? Bacteria require larger amounts of protein to survive. That's why they are able to cause spoilage in meat earlier than that of cassava.
Okay, that's a good question. When you're talking about microorganisms, okay, some microorganisms will need more of protein than carbohydrate. Others may also need carbohydrates than protein. So it depends on the type of microorganism that is going to be present. But generally, most microorganisms will feed much on protein as compared to carbohydrate.
Okay.
Elizabeth.
Is
Thank
that
you.
okay?
Yes, please.
Okay, so that is it. Now let's move on to.
Sir, because of the noise, I didn't get you clearly.
Hey.
With the explanation you just gave, please.
The question or to the question I asked?
No, to the answer you gave right now.
The answer I gave to her question.
Yes, please.
I was saying that she says that doesn't mean that microorganisms need protein, only protein for their growth. And I'm saying that no, it is not like that. Some microorganisms may feed better or may grow better in the presence of carbohydrates, okay, or other nutrients. But generally, protein will be the key nutrient for most microorganisms to grow and then what? Multiply. Is that okay?
Okay, thank you, sir.
You're welcome. All right. So let's move on to look at the pH and then organic acid. When you talk about pH talks about the acidity or alkalinity of a substance, okay? So it is looking at how acidic or how alkaline the food item or whatever you are considering is. Now, yeast and mold are acid tolerant than bacterial. So some food have low pH because of inherent acidity. For example, fermented products, because of the lactic acid during fermentation. So you realize that when you are looking at the acidity, the pH, we are saying that the pH of a food item or the organic acid presence in the food item can influence how microorganisms grow. Let me just give you a specific example or a typical example again. Take orange, okay? You as an individual, when you take orange, how do you feel? Orange that is not matured, is not grown. When you take it, how do you feel? It's sour, right?
Yes, sir.
Yes, please.
Yeah. So if you plug that orange that is sour or the orange that is not matured, grown, and then you keep it in a room, and the orange that is very grown, ripe, and then you keep these two in the room, which one do you think will get spoiled easily and why?
The ripe one.
The ripe one.
The one, the ripe one will.
Who is talking? I can't hear you.
Hello, sir.
Yes.
Sir, please, the grown one will get
easy
spoiled.
Why? Catherine, give us the reason.
The matured one will get spoiled easily than the unmatured or the other one, because the one that is not much grown, the acidic content is very high in it as compared to the grown one.
Okay. So if the acidic content is very high, what will happen? What does it do to the microorganism?
Says some microorganisms cannot survive in the high acidic content.
Okay.
Uh-huh.
So when the place is acidic, some microorganisms cannot survive there. So if those microorganisms are present, indirectly, they cannot do anything. So they cannot get the food spoiled. Okay. Then you look at, they are saying that yeast and mold are acid tolerant than bacteria, which means that if yeast and mold are present, they can actually multiply regardless of the presence of acid. You get it? If these yeast and molds are present in the food item, regardless of the acidity, if there is nutrient and water activity is okay for them, they will still multiply. Is that okay?
Yes, sir.
Then we talk about water activity. As you know, we all know that even human beings, if you don't take water, you dehydrate, right? The same way, if there is no moisture in the food, if there is no water in the food, your hand has been absent since.
Hey.
If there is no moisture or low moisture content, you realize that it slows the growth of microorganisms. But if the moisture content in the food is very high, it will fasten or it will quicken the growth and multiplication of microorganisms. I hope that is clear.
Yes, sir.
So they said yeast and mold can tolerate lower water activity than bacteria. Gram-negative bacteria require higher water activity than gram-negative bacteria. So the yeast and mold, that is why if you remember, you see that something like bread, okay? Bread doesn't have much moisture in it or water in it. But if you leave bread outside for a very long time and in a rubber, you pick it and you realize that molds are growing on it.
Yes.
So it tells you that with or without water, mold will still grow in an environment. But some microorganisms, such as bacteria, cannot grow in the absence of water because they will be dehydrated. They cannot grow. They cannot do anything. And we are saying that the same way, gram-positive gram-negative bacteria will need a higher amount of water activity than gram-positive bacteria. Don't ask what gram-negative and what gram-positive bacteria are. I'm giving it to you as something you should go and search for and read on it. So today I'm giving you two things to read on, right? I said you should go and look for the shapes of the shapes of bacteria and the names that can be given to them based on their shape. I've also given you, go and search for what gram-positive and gram-negative bacteria are. I've given you two things to go and do. Let's move on. After the water activity, we are going to talk about the biological structure of the food item. So the biological structure here, we are talking about the makeup, how the food item is actually made up or is designed. Now you realize that every organism or every food item or everything in their natural state, they have certain coverings that protect them from the environment. So you can take human beings, you can take food items like fruit and vegetables. Everybody has a protective layer that protects the cells or protects your body from invasion of microorganisms. Lawson, please, I'll call you. Let me finish with this explanation. I'll call you. So, for instance, when you take fruit, you realize that every fruit has its own protective cover. Orange has the rind, okay, that protects the orange. You talk about apple. Apple has this rubber-like, how do you call it, skin that protects it. So what happened to apple if you cut it and then you expose the inner part of the apple to the environment? What happened to orange if you cut it and you expose the inner part? If you don't cut these food items, you realize that they have their own protective covers that is covering them or protecting them from the environment. So microorganisms cannot actually feed on them. But immediately you expose the inner part to the environment, microorganisms set in, and then they start to what? They start to work. That is why if you leave a food item there, which is not bruised, is not crushed, you leave it there, it can stay for a period of time before it goes bad. But if that food item is crushed or bruised, you realize that it will not keep long. It will start what? Deterring or deteriorating. Is that okay?
Yes, please.
All right. So you take something like milk, for instance. Milk has no protective cover and it is very rich in protein. Milk is rich in nutrients. So because it has no protective cover and it is rich in nutrients, if you have fresh milk from the cow, milk fresh, you leave it from morning to evening, you realize that immediately it will start fermenting. It starts fermenting because microorganisms are there, and then because you didn't do anything to it, they are active, they start working. The nutrient is there present for them. There is moisture, there is the temperature there is okay. So they will start to act on the milk. Is that okay? Now let me come to Lawson. Lawson, please, your question.
Add a little.
Okay, Mr. Emma, let's take Lawson's question, then you add that, please.
Okay, so sir, please, actually, mine is not a question, but I'm just raising concerns. Okay. It looks like all your examples that you are giving is on food and nutrition. Please, can we also have some examples on textiles as well?
Yes, we can have examples on textiles. So let me go back and then give textile examples, okay? So we are talking about nutrients. So the nutrients we mentioned, for instance, please, a minute. So for the clothing, for instance, as I started, I said that the fabric that is being used, you know, there are certain fabrics that where they are produced from, if they are produced from animal sources, you know that the animal sources have some nutrients, okay? The same way, if it is from plant sources, the plant sources also have some nutrients. So the fiber you are looking at, for example, if you take a cotton shirt, a pure cotton shirt, and then you leave it at a place where there is moisture, or the shirt is like you wash it, you didn't dry it, and you leave it for 2-3 days, what do you perceive in the dress? What do you see?
Hair not sent to.
And most starts say bon, it will start developing smell. What else?
You see
It
black.
develops mildew.
Yes, let's take it one after the other, please.
So sometimes the color
Yes,
also
Sarah.
changes.
Sir, it will develop
It's
mildew.
discolors. It will develop mildew, which is an example of what? Fungi, right?
Yes.
Let's continue. So,
hello, sir.
Yes.
Sir.
Yes.
Please, I want to get something right.
Okay.
Please, is it always that the bacteria, the microorganisms always from somewhere get into the food, or some are already in the food,
some
causing the fermentation or whatever?
Some may already be in the food because we say they are everywhere. They are everywhere in the water, in the environment, beneath the water, even within you as an organism, within you as human being. Microorganism can be inside of you. So they are everywhere. So they can be in the food, and also they can settle on the food later. You get it? So that is why, for instance, when you are cooking, are you a clothing student or a food student? So that I know the example to give.
So I'm a food student.
Yes. So when you are going to cook a food item, for instance, any food item that you are not going to apply heat to, you are asked to either wash with vinegar or solution. Are you with me? So as a food student, any food item you are going to cook without applying heat to it. For instance, if I'm making salad, I'm not going to apply heat. So I'm supposed to wash the food or the vegetables I'm using to prepare the salad with vinegar or salt solution. What is that going to do for me? That is going to help me destroy microorganisms that are present in the food. Some of the food, some of the vegetables you even use, you blanch them, which is also helping to do that. If you be cooking the food, you would have to apply heat to it. And all those things are ways of what? Destroying microbial activities or microorganisms that are already present in the food. Now, after that, you are asked not to expose the food to the environment, because if you prepare it and you expose it, microorganisms can resettle on the food. That is what we call recontamination. Is that okay? So the microorganism may be there, but during cooking you may destroy it. But if you leave the food or you do certain things that are not supposed to done, that are not supposed to be done, you will recontaminate the food again. Is that okay?
I mean, you have exposed the environment.
I'm not getting what you are saying, please.
I said, when you cook and leave the food like that, then you have exposed the food to the environment.
Yes, please. Please, are you okay now? The person who asked the question. The person who asked the question, are you okay? Hello?
Yes, sir.
I think the person is so quick. Oh, hello.
Mr. Lawson. Yes. Mr. Lawson. Please, I hope your question
sir.
has been answered as well.
Yes, sir. Yes, sir.
You're welcome.
Thank you.
So once again, Mr. Lawson, let's look at this one. So with your part, when you are talking about the biological structure, for instance, if you are talking about, we are looking at fibers, the material you use may not have what do you call it? May not have this biological structure as we are making it clear for you to see that, okay, maybe if it is in fruit, the fruit has a skin that is protecting it. But that is why the fiber, like the material, when they produce it, they also keep it in the rubber. Okay, keeping it in the rubber, they don't keep it moist in the rubber, they keep it dry to make sure that microorganisms do not go into the fiber. Sometimes you even use you see the what do you call it, the colors they use in dyeing the cloth, or they use for the coloring of the cloth, serve as a protective cover for the cloth, because you know they are chemicals, right? Mavis, please mute.
Hey. Sorry.
So there are also chemicals which will prevent the cloth from being exposed to these microorganisms or from microbial invasion. Okay, now let's move on. I hope nobody is having any problem again. Zura, your hand is up. Zura, we can't hear you. Okay, let's move on. So we talk about.
Sir, please, I was talking to someone. Sorry.
Okay, so we have mentioned about 4 internal factors that we said they are intrinsic factors. We mentioned nutrients, we mentioned pH, we mentioned water activity, and we also mentioned the biological structure of the food. I hope you have not forgotten. Then now we are going to look at the external factors. Now, when we talk about, before we move to the external factors, when we talk about the internal factors, you know, these factors are all within the food. So the nutrient we spoke of is within the food, right? The moisture we are talking about is within the food. The pH is also within the food. The biological structure is form part of the food. You get it? So all those things that form part of the food are what we call the internal factors or the intrinsic factors. Now let's move on to look at the extrinsic factors. These are factors that are external, or they are found in the surrounding of the food or the cloth. So the external factors or the extrinsic factors are factors that are found at the surrounding of the food or the cloth item. So for instance, relative humidity. When you talk about the relative humidity, it just looks like what you call the water activity. But in the relative humidity, we are looking at the amount of moisture or amount of water that is in the environment, not within the food, but in the environment in which the food is being kept. Is that okay?
Yes, sir.
Hello. Are you getting the difference?
Yes, sir.
Yes, sir.
Okay, so the amount of moisture that is found in the food environment, and you realize that anytime you keep food or you keep clothing at a moisturous environment, an environment that has a lot of moisture, what happened to the food or the cloth? You realize that the food item will start getting spoiled. The same way the clothes will also start developing certain things, characteristics like smell, change in color, especially if it is white or any plain color that you can identify. You realize that there is a change in color and all those things because there is moisture in the environment. And immediately there is moisture in the environment, microorganisms are able to freely move, freely express themselves. So that is about the relative humidity. It says that they increase the water activity of their own immediate environment so that eventually microorganisms requiring a higher water activity are able to grow and spoil food, which was initially considered to be what? Microbiologically stable. The same way in clothing. So what you are trying to say is that when there is moisture and these microorganisms are present, you see that physiologically they become active. And after they are active, they'll start respiration, they'll start multiplication, and then they are looking for nutrients. So if there's nutrient for them and then they get hold of it, now they start to multiply and then they start to exhibit their characteristics. We move on to look at temperature. Temperature, we say it is the hotness or coldness of an area within a specific period of time. So microorganisms grow at different temperatures. Based on the kind of temperatures at which microorganisms grow, you can have 4 different groups of microorganisms. I think here we have 3, but then you have 4 in other books. So some microorganisms grow at a very low temperature, where the temperature is very low. So such microorganisms, we call them psychophilic microorganisms or psychophores. We call them psychophilic microorganisms or psychophores. So psychophores can grow at a temperature of 1 to 15 degrees Celsius with an optimum temperature of 7. You see, and if the temperature goes beyond 20, psychophores will not grow. When we come to the other group, we have mesophils. They are also microorganisms that are able to grow from the temperature range within 20 to what? 45. And you realize that this is the temperature range that we call at room temperature. So it says that they have an optimal temperature of around 27, what we call the body temperature. You know that the body temperature is supposed to be somewhere around 26, 27. Please, are you aware of that?
No,
yes,
sir.
please.
So room temperature is actually considered as 25, and then the body temperature is actually considered to be around 30, 36, 37. So if you go to the hospital and they check your temperature and it goes beyond that or below that, it means there is something wrong. You get it? Not always that there's something wrong, but then it will raise question to sometimes maybe there's something going on that we are supposed to look at, because we consider that a body temperature should be a range. Mostly some people give range like from 35 to 37 is a normal temperature. But if it goes beyond that, then there is a problem. Or if it goes below that, then there is a problem. So we have microorganisms that can also grow very well at this temperature. And we call these microorganisms mesophils. We call them mesophils. And you realize that most of the foodborne pathogens that cause food spoilage and then spoilage in clothes and all those things are all mesophils. So we say that these are microorganisms that grow at room temperature. They are called mesophils. Then we have thermophils or thermophiles. Thermophils are microorganisms that grow at extremely higher temperature. So they grow at an environment where the temperature is extremely high. So for instance, people in production of milk, when they are doing the pasteurization of milk or sterilization, you realize that it is done within so pasteurization is actually application of heat to milk within a short period of time, after which it's cooled. If you should leave that food item and it is heated beyond or above the temperature at which it's supposed to be, you end up introducing the growth of different group of microorganisms in this food item. So temperature is very important. That is why mostly you are advised to boil or heat your food item when you are not going to be taking it, or you keep it in the fridge if you want to store it for a longer period of time. The same way for those in clothing, we say that you don't store your clothes in a particular temperature. You store your clothes at a place where the temperature is extremely high, like in the oven, or extremely high. I don't know. some rooms can be high, but not to that extreme. But storing clothes at an extremely high temperature or extremely low temperature can also influence the clothes. So sometimes you store your clothes at a place which is very hot, and then for a very long time, one day you just go and pick the clothes to wear, the dress, you pull it like this, and then you see that everything is tearing because the fibers and things have become loose. Okay, so it will tear. That is what happens. Then we talk about the gaseous environment or gaseous environment. The gaseous environment talks about the amount and then the type of gas that is in the environment. So you realize that in food production, certain foods are being stored by putting in carbon dioxide. So something like the fizzy drinks, like those Bigo Cola, Coca-Cola, and all those things. Those ones that when you open, you hear the bubbling sound and all those things. They have been preserved using carbon dioxide, which prolong their shelf life. And then they make sure some of the food items are also stored in an oxygen, the word is not coming. In an environment where oxygen has been used to, nitrogen has been used to substitute oxygen. You know, oxygen is when air is available, plenty air, excess air is available. It also influences the growth of microorganisms. So oxygen is being substituted with nitrogen. So we say that there's a name for it. It's nitrogen. Oh shit. The thing is not coming. Oh, come on. Is somebody helping me? Yes, is somebody helping me?
Sir, please come again.
Come later.
Is it?
It will come later.
Also?
Once you are done with the class, you see you remember it. So don't worry.
Yeah.
Please come again.
Okay, so let's continue.
With what you
I was
asked.
just looking for, like, I'm saying that the exchange of air. So you talk about gaseous exchange, right? So where nitrogen is used to substitute oxygen. So there are rooms that are nitrogen enhanced rooms. So oxygen is being driven away by the introduction of nitrogen. And due to that, it's helped the food item to be stored for a longer period of time. Is that okay?
I searched and they gave me nitrogen hypoxia.
Nitrogen what?
Hypoxia or nitrogen anosmia.
Oh, okay. It is correct though, but that one is big. It's correct. It's not wrong. It's correct.
Okay.
Yeah. Okay. So let me do the last slide and then I'll close you because I know that you are going to join a different class. Even this one, I've done it already. I've done, look at, we are moving on to look at fiber, but we're doing it hand in hand, right? Hello.
Hello, sir.
Hello, sir.
Yeah, I was saying that we're going to look at the factors that influence microbial growth in fiber, but then we're doing it hand in hand. So,
yeah, please. About the oxygen.
About what?
The oxygen. After the gaseous environment, you have the oxygen.
Yeah.
And the ground positive.
Yeah. So oxygen is the most important gas in contact with food. So I'm saying that when there is oxygen in the environment, a lot of oxygen in the environment can cause food spoilage. You don't know. That is why they always tell you that put your food in an airtight container. Those in food, when they are telling you how can you preserve this food for a long time, they say that put it in an airtight container, store in an airy place. Do you remember?
Yes, sir.
Yeah, so those are some of the things. So please, to those including, I'm not leaving you out, but then when we're doing it, you realize that I was explaining hand in hand. So let's pause here. Just try to read. Now, it is not only for this slide, it's not only for those including.
The modified atmosphere.
Atmospheric environment.
Yes.
That's what I was looking for. Thank you very much. Okay. It's not easy. Sometimes when you're remembering something and it's not coming. Mr. Ima, thank you. So please, let me pause here. Read on this so that when we meet next week, we'll just try to summarize it. Then we'll do the last one, and I think we'll be done. Is that okay?
So please, are we meeting next week?
I thought we agreed on that. Or?
We have. Yes, we have.
Next week is quick assessment.
Somebody is raising a concern. Please, what are you saying?
We are meeting.
So she said
So we are meeting.
she thinks next week is assessment.
We are meeting.
Assessment is
Assessment is 22nd. It's 2 weeks from now.
Practical, so 15th to 16th.
We've talked about this. It means that you were not here earlier.
I think last week we discussed this before we agreed on meeting last week.
We've discussed it already.
Yeah. So
she was not around earlier.
So thank you very much. Try to read more and pray that I'll get time so that I can see if I can add another set of questions for you to.
That will help. Okay, so
yeah, pray.
Okay, so thank you for joining, and then steady hard. Make me proud.
Okay, sir.
Thank you.
Thank you.
We'll do that.
Thank you very much, sir.
Yes,
madam. You don't have the slide.
Yes, sir.
Yes, sir. Yes, madam.
Please, sir, there are something to give. Some of us didn't get.
Assignment. He said. Please, are you listening?
Well, man, just put it on a platform.
Listening. It's not an assignment. I think he said we should go and read on it. Or
it's assignments, but not for submission. It's not all assignments that are for submission. So with this, you will find out when we meet. You'll be asking. So if you get 2 or 3 people who answer correctly, it means that we have done it, and then we move on. But not assignment that we will write and submit. You get
it? Give it to us. Exactly.
So I'm just saying it. This is not to submit for me to put it on a platform. This is, he said we should look out for the various shapes of bacteria and then the names that we give to them. And then he said we should also look for the gram-negative and then the gram-positive bacteria, what it is at all. You see?
Gram or gram?
Hello, Saima.
Yes.
Please, the science slides, is it up to 6? That's unit 5, or it's up to 6?
Oh, you didn't get the second question. Hello, Baima.
Yes.
Please, you said the gram negative and then the gram positive.
Bacteria. What is it? So this thing, within 10 minutes, you can find out this thing. So next week we come and then you will just present it and then we are gone.
Say, you have not answered me. I said the flies. Is it up to 5 or 6?
It's up to 6.
Then we have not received the 6th one.
The one I put it on, is it up to 6 or 5? The one I compiled.
It's up to 5.
Oh, let me cross check again. I think it's 6. If it is up to 5, then I did not add the 6th one. So you
It's
let me
up
check.
to 5, Saima.
Then I will check. If it is up to 5, I will check. If I add the other one, I will put it on it. Okay, so let's go and relax, and then we have 25 minutes to the next lectures. 11:50. Okay.
So
Bye
please,
bye.
the next lecture will be what?
11:50. Foundation.
Okay. Thank you, sir.
Okay.
Say, if this week we will do discussion, please alert us, those on different centers. That's group discussions.
Okay. Okay.
Okay.